the next generation of medical innovations. Over the next few hours, you'll get to meet the startup founders that are turning science fiction into reality, as well as the investors and partners that are giving them the resources that they need to grow. To start with, I invite you to take a moment to think about the lands that we're meeting on and the people that have brought us here. We're meeting on the lands of the Bidjigal people, and we pay respect to them as the traditional custodians of the land, as well as their elders, past and present. People have been meeting on these lands to share stories of knowledge and inspiration for tens of thousands of years. And it's today that Horizons Ventures has brought us together to celebrate, to innovate, and be inspired. I want to acknowledge and pay respect to Selena Chow and Debbie Chang for their leadership and philanthropy that makes today possible. You're going to see panels, you're going to see people talking, there's a great expo outside. If at any point you're not sure where to go, what to do, how to get where you want to be, talk to anyone in the black shirt that has founders on it. They will be delighted to help you. You're going to hear a lot about the word innovation, a lot about the word investment, but the thing that unites those two concepts is what the University of New South Wales and Horizons Ventures both believe, which is the power of philanthropy. I'm David Burt, I'm the Director of Entrepreneurship here at the university, and we use the power of philanthropy to find and nurture the next generation of entrepreneurs from the university community. And representing UNSW, and to formally open the event, I'm just about to invite up to the stage Professor Bronwyn Fox, who's the Deputy Vice Chancellor here at the University for Research and Enterprise. That means that Bronwyn leads a team of over 700 people that are the research and commercialization infrastructure of the university. More importantly, if there's a technology that matters in Australia, Bronwyn has a hand in guiding it. She was Australia's representative to the International Panel on AI Safety, sits on the strategic advisory committees at the federal level for both robotics and quantum, and personally is an internationally celebrated expert at material science. Just to make it sound even more impressive, also has an Order of Australia, which is one of Australia's highest honours, for distinguished service to scientific development and research, advanced manufacturing, and tertiary education. Please welcome Bronwyn.
Thank you, David, for that very kind introduction. Hi, everyone, and welcome. It gives me great pleasure to welcome you to the Techcracker Lab, collaboration between UNSW and Horizons Ventures that brings together some of the world's most innovative minds to inspire our next generation of entrepreneurs. I'd also like to begin by acknowledging the Bidjigal people, the traditional custodians of these lands, where we are fortunate to have this amazing UNSW campus. We're privileged to work, learn and study at this place where knowledge has been exchanged for tens of thousands of years. I pay my respects to elders past and present and extend that respect to all Aboriginal and Torres Strait Islander people joining us here today. Today, I'm emceeing on behalf of our Vice-Chancellor and President, Professor Attila Brungs, who is extremely disappointed not to be here. He sends his warmest regards and best wishes for what promises to be an extraordinary event. I'd also like to extend a very warm welcome to our distinguished guests. Senator Dave Sharma, Shadow Assistant Minister for Competition, Charities and Treasury, our Chancellor, David Gonski, ACE here at UNSW, who's also Chairman of the UNSW Foundation Board. Ms. Selena Chow, welcome. We are delighted to have you here, the co-founder of Horizons Ventures and co-founder of the HS Chow Foundation. And Ms. Debbie Chang, co-founder of Horizons Ventures and co-founder of the HS Chow Foundation. Welcome. Thank you all for joining us. Your presence here speaks to the growing momentum behind Australian innovation, and we're thrilled to have the opportunity to connect and collaborate here. It's fantastic to see such a diverse audience here today, from students to alumni, researchers, founders, and industry partners. The breadth of experience and perspective is what makes events like this so dynamic. So the purpose of today is twofold. First of all, we are here to showcase some of UNSW's leadership in medtech, research, innovation and entrepreneurship, to give you a glimpse, just a taster of the exciting work that's happening here. Second, to set the stage for collaboration. This is a rare opportunity to connect our startups and students with global investors. So I encourage you today to be curious, ask questions, and to make new connections. We are so proud to co-host this event with Horizon Ventures, a firm whose global impact speaks for itself. Horizon has backed some of the most transformative companies of our time, including Meta, Zoom, DeepMind, and Spotify. Absolutely breathtaking. Their mission, see problem, solve problem, repeat, aligns so powerfully with our own strategy here at the UNSW, which is progress for all. At UNSW, we believe that great research doesn't just change minds, it catalyses the creation of entire new industries. It changes communities and it changes lives. And that belief is actually backed by action. In 2024 alone, 19 new spin-out companies were launched from UNSW developed technologies, tackling challenges across solar energy, virtual healthcare, advanced mapping, and more. Through initiatives like UNSW Founders Barker Street Ventures and our Entrepreneurial Campus program, we're building an ecosystem where science meets impact and where bold ideas can thrive. Thank you so much to all of you for being here today. I hope today sparks new ideas, new partnerships, new connections and new possibilities. We really have an action-packed agenda today, and our first session will begin with a panel focusing on lessons from tech investors, and then we'll hear from our three sets of keynote speakers representing Harrison AI, SynthMed Biotechnology Incorporated, and HoloClara. Then we'll have a short coffee break where you can check out the MedTech Expo. And now it's my pleasure to hand back to David, who's going to introduce our first panel. Thank you.
Some companies you can create with your own time and initiative, maybe with customer revenue very early in the journey. For every other type of company, and for the most ambitious ones, you're probably going to need an investor on day one. So the purpose of this panel is to hear directly from the people that have really done the most in Australia to support the type of ambitious companies that we're here to celebrate at Techcracker Lab. So to start with, I'd like to invite Bill Barty up onto the stage. Bill, round of applause. Bill doesn't know this, but I regularly get asked by founders. We help hundreds of them here at UNSW every year. I'm regularly asked by founders, who's the best investor in Australia? I don't know, but the answer that I tell them is based on the investor that when I ask founders, who do you want as your investor? Who do you want on your board? The name that comes up the most is Bill Barty. Bill has done an incredible amount, not just to fund the companies, to write the check, but when it gets hard, Bill's the person that's there and is supportive. And when I ask people, why do they say that? It's like, Bill reacted the way I wish all my other investors reacted. Bill is the founder and managing partner of Main Sequence Ventures, which was born out of the Commonwealth Science and Industrial Research Organisation. He's also co-founder and general partner at Blackbird Ventures, among many other things. Just so he doesn't get too big of a head, not so great at running and swimming, but those are his passions outside of work. Next, I'd like to invite Emerald Schofield up to the stage. Please welcome Emerald. Emerald is an associate at Main Sequence Ventures, focusing on backing deep tech companies in health, things that are going to feed 10 billion people, help decarbonize the planet. Great example of the talent we extract from New Zealand. Prior to joining Main Sequence Ventures, worked in deep tech venture capital in the New Zealand market. Really grateful to have Emerald with us on the panel, an expression of the type of talent that is here walking around our university, building relationships with researchers and finding the next deep tech company. Next up, James Cameron from Airtree Ventures. Please welcome James. Graduate of UNSW, James started life on the founder side of the fence, took that company to America, and then as part of some of the relationships and skills he was building in America, joined Accel Partners, one of the sort of best venture capital firms in the world, before returning to Australia, and he's now a general partner at Airtree Ventures, where he has backed several UNSW startups, including High Earth Orbit and GridSight. Please welcome James. Finally, we have Chris Liu from Horizons Ventures. Please welcome Chris to the stage. I think Chris's best quality is I feel he is eternally curious. I've had the pleasure of asking some people that sit on boards with Chris or has taken some money from Chris, and they describe him as if he's interested in it, he is intensely interested and focused, super present. And together with Main Sequence Ventures, Chris has been involved in backing some of the most ambitious companies in Australia from incredibly early in their journey. And so obviously with the rest of the Horizons team as support around him, there are several companies, the really powerhouses of Australian industry. I'll pick on Cauldron Fermentation. Michelle is here in the room. That is an amazing company that Michelle is building on behalf of the world. Would not be possible without some of the partnerships between Chris and Main Sequence Ventures. That's it from me. Over to you, Bronwyn.
Thank you so much. That should be working now. Excellent. Yeah. Thank you, panel. Thank you so much for joining us today. We really appreciate it. Bill, I'm going to start with you. You've been described as a founding father of the modern venture capital scene. You can blame David for that description. But I'm just wondering how you've seen things evolve over your time and what the single biggest misconception that founders have today about securing capital compared to when you started, and how's that changed?
Cool. Thanks. Thanks very much. Well, first of all, before we start, I would like to thank Selena, Chow, and the entire Horizons team, as well as UNSW, for inviting us here today. This is a terrific opportunity, I think, for all involved, so thanks very much to everybody. Well, I think one of the things that's changed is 25 years ago, we wouldn't have a gathering like this. There would be no opportunity or no event like this. There was very few sources of capital. That was despite the fact that at the university level, there's always been very, very strong research in Australia. But what was missing really was the ability to sort of work together with the academic community and turn some of these great ideas into real companies. And I think one of the things you're going to see up on stage today is you're going to see a lot of that in reality, because the thing that you'll see behind us is going to be pretty mind-blowing, I think, and there's several others up here that will be pretty amazing. But what's happened over the last sort of 25 years is that we've had investors come into the market to be able to recognize the opportunities that are here. And I think sitting up on stage, some folks who are starting companies up may not think that there's very much capital around, but there's between Airtree and Main Sequence and Horizons, there's lots and lots of money around, but you've got to have a really great idea. And so even though there's capital, you've got to have a terrific idea that has impact and ability to drive returns. So what's changed is that, but even more fundamental, I think the most fundamental change I have seen is that in that period of time, the attitude has changed. And so it used to be that students coming out of universities wanted to go to work for a law firm or an accounting firm or some big organization. Now they want to come out and be entrepreneurs. And that's a grassroots change of attitude, I think, that's been very, very beneficial for Australia. So I hope that continues, because we're ready to meet you and ready to back you if you've got a terrific idea.
So how do you think that plays out for the expectations of founders? Do you think that maybe there's more opportunities than they expect?
Look, the opportunity is only as big as their imaginations. So I would say if you're doing something that's impactful and you really want to go for it, there's plenty of people in the room and up on the stage that are willing to listen and be able to be your partners here. So.
And since you've pointed at Emerald, I'd like to go to you for the next question because you represent the next generation. What are the misconceptions that you are busting, and what are the things that where you're seeing change in particular?
Look, I think the key thing that I would say is deep tech investing, or just deep tech in general, is not the same as tech investing. And so ultimately, while they're based on the same principles, they need to have a very strong problem and market pull and fantastic founders to actually build the solution. With deep tech, you need to have that and more. You've got a very complex marketplace that you need to navigate. You need government policy, and you need to have corporates and customers that are willing to step down and help you earlier. Whereas, like, when you look at a SaaS startup, like a problem on their dashboard, for example. Whereas if you're looking at a deep tech startup, you are, quite an expensive thing on their balance sheet. And so while you're looking at something that's got totally different timelines, different capital constraints they're looking to unlock, and then also a very different risk profile. And in saying that, it just means that you need more patient capital and collaboration to make it work. And so I think the thing that's been really exciting about watching the deep tech ecosystem here, and actually comparing it to New Zealand as well, is there's so much lean in, not only from investors, but from government and industry. And so I think realistically what we're seeing now is, while the first generation of kind of deep tech investing has kind of been in the last 15 years, was bridging the gap between the first valley of death of commercializing research out of universities. What we're now seeing is a maturation of technologies and companies that are now facing the second valley of death, which is showing commercial proof. And so the thing that we're now having to bust down is getting funding for those companies, helping them get corporates engaged earlier, and essentially also build a new industry in a new environment. And that takes time, market annealing, and a lot of effort, and some really courageous founders who are putting their time and effort on the line to do so. And I think ultimately, like, this industry is really on the precipice of, like, really establishing itself in the Australian ecosystem, and with success breeds success. And so I think it's an absolute privilege. Like I always say, I have the best job in the world, and I get to work with the coolest people on earth. And so we're potentially about to see a lot more success cases that are going to really make Australia stand above and beyond on the world stage. And so that's really what we're busting, and it's like a really great job to be working alongside people doing that.
Thank you so much. James, over to you. Airtree's had a focus on software as a service, and we had a chat earlier, and you've got your own background is in computer science from UNSW, very proud alumni. I'm just wondering, given this trend into higher risk tech investment, would you see there being an intersection there that you can exploit at Airtree with cyber physical systems?
Yes, well, thank you firstly, Bronwyn. It's brilliant to be here. I am a very proud alumni. It's great to be back on campus. I was reflecting on the walk up the campus about my 2 years at residential college here at UNSW, at Baxter College, being 2 of the most formative years, I think, of my life. And it's absolutely brilliant to see the generosity of Selena and the Horizons team, and part of that grant coming into residential college. It's really a wonderful donation, so thank you very much. And just touching on a point that Bill made, I think the last time I was in this very room was probably about 30 years ago at a careers day. And at that careers day, there was all the usual suspects 30 years ago that would have been hiring UNSW grads. There was a line out the door for Goldman Sachs, and there was another line out the door for McKinsey. And of course, the startups didn't exist back then. Deep tech, software, nothing really existed back then. And the biggest and most exciting change, I think, in the 30 years since I was a student here, is that these days, when I come back to career events here, there are lines out the door for some of our startups like Canva and Go1 and Employment Hero, and it's fantastic to see the enthusiasm that a lot of these startups are seeing from students who are coming through the ranks. As Professor mentioned, we focus mostly on software. I think I've always struggled a little bit with this dichotomy between SaaS and deep tech. We don't use the term SaaS quite a lot internally as VCs. I think SaaS for us is a business model. It's really just selling software on a recurring revenue basis, and there's very few software companies these days that don't try and get some sort of recurring software on it. So it's not something that we would use to categorize any of the investments we make. But I think really what we're talking about is this dichotomy between software and deep tech, which I hear a lot, and even that I sort of struggle with a little bit, because I think we would all admit that some of the most exciting advancements over the last 4 or 5 years, certainly in technology generally, have sat within the software field. I think you just need to walk across the road to my old department over at the Computer Science and Engineering School and ask them what they're working on and whether or not they consider it deep or shallow tech, and I think you'll probably get a few blank stares for the question. But if we thought 5 years ago that we would suddenly be here in 2025 with 4 or almost 5 research labs globally who have GPT-5 class foundational LLMs who can pass the Turing test without breaking a sweat, I think none of us would have imagined a world like that. And the technology and the advancements there are really digital technology and software. And that's what we focus on at Airtree. It can run the gamut from developer tooling to AI to cybersecurity. I personally focus on a lot of the sort of unsexy part of the software stack that I find particularly exciting, but a lot of people find a little bit boring. It's the data technology, developer tools, cybersecurity, AI. Again, 5 years ago, that was very unsexy. Nowadays, suddenly it's become sexy, which is great. But one of the things I liked the most is now that there is this emerging categories of venture capital that really can apply to so many different fields within the technology landscape more generally. So many of our founders are learning from founders across the board, be they starting hardware companies or health tech companies or software companies. And I think it's important because I think for all the founders out here in the room, you will know that starting a company is the hardest thing you can do professionally. You realize pretty quickly when you start a company that everyone is out there trying to kill you. Your customers want to pay less, your competitors want to wipe you off the face of the planet, even your employees want to earn more and drag down your cash runway. So everyone's trying to kill you. And the only thing that you can do to counteract that is increase your speed of learning. The best founders that I've worked with will always increase their speed of iteration and learning. And the best way that you can learn is by talking to other founders and understanding some of the challenges they've had. And this mix of deep tech and hardware and healthcare and software startups now learning from each other and learning from some of the benefits and challenges that each of these individual models have is an incredible part of the Australian ecosystem that didn't exist when we started Airtree 10 years ago.
Thanks, James. So Chris, James talked about that dichotomy between deep tech and SaaS. Of course, Horizon Ventures operates globally. How do you see that playing out globally? And what do you think is unique about Australia? What's distinct about what we do here?
Thanks, Ron. I get asked this a lot, and just as James said, I think Horizon's been investing in Australia since, it was about 2017 now. So we've kind of been part of this whole ecosystem of watching this industry mature over time. And I think some of the observations and learnings I've got, number one, from a software perspective, there is actually scale now. And this doesn't matter whether it's deep tech or not deep tech. I know Canva gets mentioned a lot, Safety Culture gets, some of these bigger names. But what Australia often forget, that you've got tremendous amounts of other software companies that are very large, like Seek, who's an employment, who's a jobs HR company, and then there's realestate.com. These are huge, right? And also, I was just having a chat with Bill earlier at the origins of Google Maps, the team, the people that started it, they were in Sydney, I think, right?
Yeah.
And Bill knows them very, very well. But the thing is that back in those days, the Google Maps team got bought out by Google. Australia doesn't have a Google to buy any of these companies. And so there hasn't really been any companies of scale on the software side that absorbs these, right? We're starting to see that happen. A lot of the larger software companies are starting to see that because you cannot have a venture system without having an exit market, because that's sort of how venture capital works. But on the harder end of town, let's call it the deep tech, hardware, manufacturing, it distilled in its very early infancy. I think Emerald and Bill, we sort of have quite a few companies together, and what I've learned is, and I think I'll give you a couple of figures, right, is that the share of manufacturing in terms of GDP in Australia back in the 1960s, I believe it was about 25%. To give you a bit of comparison, China at the moment, I think it's roughly about 25%, depending on which figures you look at. Australia's declined down to about 15% in the 1990s, and then now we're sitting at about 5% and declining. And so when you make a decision or you're trying to get a company to work, well, you kind of have to make a decision on whether you're actually helping a company or you need to help to develop an industry from scratch. I don't know, Bill or Emerald, you want to just add a little bit to that?
Yeah, I mean, look, I think that's right. If you look, I think one of the things that needs to happen, and that is happening, is that if we're going to build industry, you have to think really long term. And if you think long term, then that gives you a chance to actually put some foundations in place. And it's also key to be able to work with the fundamental sciences and the fundamental engineering that's working. So here at UNSW, for instance, there are two companies, Dirac and Silicon Quantum Computing, both of whom have a good shot of developing quantum computers that actually work and are good. You've got an amazing physics department with some top professors, and it takes that combination of that kind of fundamental research over a long period of time, combined with capital and business know-how, to be able to put things together to create these companies of value. And again, I'm really excited to see what we're going to see behind me. I can barely contain my excitement here, but this is.
Don't touch it yet.
Yeah, I won't touch it. I wanted to pull the cover off, but I didn't. But you're going to see an example of this, where something was worked on for quite a long time at the university level and is now out in the public and is making a huge impact globally. So this kind of long-term thinking and combinations of academia with with venture and business is very important to build these long-term industries.
Just one more thing. I think that there's also we've got to pools of different capital here, right? And Australia has got very, very deep pools of capital. This is now a net exporter of capital. Yeah. And the thing is that it's concentrated, and there's only a few players that are kind of dictating the market. I think we used to James, we used to joke around and say, oh, no, the Airtree's not looking at this. Why are they not looking at it? That sort of slowed down. I mean, this is like 5-6 years ago, right? But now that's really sort of, there's a lot of collaboration going on. Everyone's trying to figure out, because I think this ecosystem needs to build a few more very large, deeper tech, manufacturing-based companies. I think that's
exactly right. I think you can see the impact that Canva and Atlassian had in the software founder landscape in Australia. It provided a blueprint on how to build a global category leader from their base in Sydney, selling to the world. And they are now 2 of the 10 largest companies that have ever come out of Australia. And they didn't exist 20 years ago. In Canva's case, didn't exist 15 years ago. So they've created a blueprint on how to go from 0 to one of the top 10 companies in Australia's history in a very short period of time. And doing that in a deep tech sense. And we've had them with Cochlear, we've had it with Resmed, there's definitely a few examples of it. But the more of them that we can get, the more that you can show that the blueprint is there, I think the better we'll have for it. And I think, just one other point on that, I think the benefit of having a much deeper and wider landscape of investor types and individuals and pockets of money nowadays is that you can create companies without having to build for what you think the investment landscape will support. And this is actually something I think the software founders can really learn from a lot of deep tech founders. There is any startups in the room, any founders or any founder-curious individuals that are thinking about funding a startup, the worst thing you can do is trying to build a startup because you think it's what an investor will back.
Yeah.
If you're taking product advice from your investors, even highly qualified deep tech investors like these three here, you're probably on the wrong path.
Yeah.
They should be taking product advice from you as the founder. And if you're building something because you think it's going to get funded, it's a great way to be in a very consensus pile. And frankly, it's probably a great way not to get funded. I recommend you are building because you understand a problem better than anyone else, and you are desperate to go and solve that problem and make it your life's work rather than doing it for what an investor wants.
Yeah, just one other thing I would say too is, the venture business has been in Australia for a very long time, except it was called mining. So there's lots of, there's been billions of dollars put into, into searching for ore and rocks under the ground, and look, that's great, but you have to expand that economy to to other things as well. The other thing that we're seeing in the ecosystem here is that, and I would describe the Australian ecosystem as very rich in a lot of ways, but very immature in a lot of ways too. But one of the signs of maturity I think that we're all seeing is what James mentioned, and that is we are seeing companies that are working and are building companies of size, and you're seeing people leave those companies and start other companies. And it's when you begin to see entrepreneurs leave other companies because they've been up that curve, they've learned some lessons, they've got an idea, they want to start their own, you then begin to, the whole flywheel starts to turn. And then when you get that, some of the founders putting their money back in, and you get groups like Horizons coming in and ourselves being able to back some of this stuff, that's when the maturity really starts. And so I think we're kind of right there. So I'm pretty excited.
It's exciting to be there.
Yeah.
Emerald, I'm going to come to you in a minute, but I've been doing some uncovering of the history of quantum physics at UNSW, and it was really catalysed by a Centre of Excellence 25 years ago.
Yeah.
So it's taken 25 years of patient support from UNSW to get to the point where we have two absolutely incredible spin-outs
Yep.
from the university. And that's not unique to us. I remember being at CSIRO and attending the launch of a number of different companies, and I'd find the scientist at the back of the room, and I'd go up and sidle up to them and say, when did you start working on this? And it was inevitably 20 years ago, every single time. So Emerald, you made a point about the power of collaboration, and I want to hear from you. I'm such a big fan of Main Sequence's venture science model. Can you explain that to the audience, given we have a number of students here in the audience?
Happy to. So I mentioned earlier around the importance of building a company that's solving a strong market problem. Like, you're not solving a problem that's a speck of dust on a customer's shoulder, it's an ice pick in their eye. It's the first thing they want to solve. And so Main Sequence has kind of pioneered this model called venture science, where essentially you work with corporates to identify a problem that they're seriously wanting to solve. And then you identify an entrepreneur who's particularly excited by that area, who will go across the research ecosystem and find the right science to solve that problem. And so ultimately what we do is we bring all of those people, so the corporate, the entrepreneur, and the research institution, to come in as founders onto the cap table, as well as ourselves as the venture founder, and we put capital in. And what we've found is that essentially by having the corporate at the table from day one, you've got a customer who's invested in the success of this company, who's essentially walking you through what do they need to see for this to be commercially viable, telling you, because a lot of the time what you find is if you're not talking to the customer constantly, you can build something that's not even relevant for them anymore, right? It's so easy to do. And so if you've got them at the table constantly going, that's not important, let's focus on this, it really makes for a massive difference in terms of the trajectory of your company, utilizing your capital more effectively and not building the wrong thing. And so an example of one of those companies that has kind of come out of the Main Sequence portfolio is Samsara Eco. They're an infinite plastic recycling company. We worked with Woolworths, and they said, look, we've got this massive problem with mixed bale waste. How do we sort this out? Because it's really hard to separate it. And so Paul Riley, who's now CEO and founder of Samsara, went to ANU and found some interesting research out of there, and we brought Woolworths onto the cap table. That company has now gone on to raise over $100 million. It's signed $1 billion offtake agreement with Lululemon. Lululemon. Yeah, and that's fantastic in 5 or 6 years now. that's really a strong speed in a deep tech startup in particular, and really leading the way in terms of how we build in this ecosystem and setting a really strong pace.
Thank you so much. This has been a brilliant conversation. Thank you all for joining us today. It's just the start of a conversation. We're looking forward to keeping in touch and to seeing you and having a chance to speak with you at the expo. I'm now going to pass back to David Burt. Thank you.
Thank you.
Thanks.
Thank you. Thanks, guys. Thank you. That was good.
Thank you.
Keep going.
So you've just heard from a group of people that collectively shepherd billions of dollars into the most exciting and valuable ideas. So if you have an idea for a company, please come and chat to anyone in a black t-shirt. We'd love to help you get started. Take a moment to think, what did you hear from this panel? What do you want to remember? There were so many different insights that were shared with you. Just take a moment to think, what do you want to remember from that? For me personally, the biggest phrase that resonated was, the opportunity is as big as your imagination. So just encourage you. One of the real purposes of today is to get you building relationships, get you meeting people that you can build with. So if you awkwardly find yourself over a coffee with someone and you don't know what to talk about, just share whatever is in your imagination. Thank you, and back to Bronwyn.
Thanks, David. It's my great pleasure to introduce our first keynote for the afternoon, and it's Dr. Angus Tran and Dr. Dimity Tran from Harrison AI. They are the visionary co-founders of Harrison AI, a Sydney-based global health tech company that uses state-of-the-art artificial intelligence and partnerships with healthcare specialists to create diagnostic solutions and help solve healthcare capacity challenges. Angus is a trained clinician and CEO of Harrison AI. He earned his Doctor of Medicine with distinction from UNSW in 2018. During his medical training, Angus identified a critical challenge facing global healthcare, the inability to scale medical expertise to meet growing patient demand. This realisation helped him to co-found Harrison AI in 2018 alongside with his brother Dimity, with the mission to raise the standard of healthcare for millions of patients every day. Dimitri is a healthcare entrepreneur and chartered financial analyst by training. He helped build and expand Harrison Analyse AI, which is now assisting clinicians in more than dozens of countries, serving millions of patients every year. Their AI-driven imaging solutions are helping clinicians to detect cancer and other conditions faster and more accurately, with a mission to raise the standard of care for 1 million patients every day. So I would like to introduce Dr. Sebastian Himbert. We're ready to go. Hello, sorry, hi. Welcome, thank you.
Earlier this year, the BBC channel covered the story of Diane Covey, a 69-year-old retired hospital worker from Surrey, UK, who's been referred by her GP to have a chest X-ray at the NHS for a bout of cough. Just like hundreds of other patients like Diane that day, she's been cleared by her care team with a normal chest X-ray being sent home. It just so happened that this is the day Harrison AI has gone live at this particular hospital. Within hours of being switched on, the AI rapidly analyzed hundreds of cases of X-ray images and identified Diane cases with something subtle, something that was originally missed on initial review. Stage 1 lung cancer. What would have otherwise been a death sentence, caught early enough to change everything. When the BBC interviewed Diane about how she felt about the fact that AI analyzed her case, she said, of course in brilliant British accent, "I never really understand AI, but I think it might have just saved my life." The fact that my cancer has been caught in the earlier stage gave me options. It gave me a second chance at life. This is the human story about the technology of our time, artificial intelligence. Diane is not alone. The math on healthcare outcome is brutal. If 100 patients are diagnosed today with lung cancer at stage 4, only 5 of these patients would be alive 5 years later. But if we just manage to wind back the clock, if we have the magic to rewind time and make those same 100 patient diagnoses at stage 1, 65 of these patients would still be alive 5 years later. The magic pill that will cure cancer, perhaps it's not a pill at all. It is the magic of early diagnosis. Early and accurate diagnosis simply save life. And this perhaps is not so surprising to many of us. The real question is, why don't this always happen? We are in the middle of a massive capacity shortage today in medical expertise. Walking this hall, I was reminded of my time as a medical student at the University of New South Wales. I spent 6 brilliant years here training to be a medical doctor. But it would take me another 9 years before I can become a specialist capable of truly provide diagnostic outcome, treatment for patients. In Australia, we are missing 1,100 radiologists and pathologists to truly meet the diagnostic need that the aging population is demanding. And that number only scales exponentially when you look across the world. We are simply not training enough clinicians to meet the demand of our future. The net effect for all of us is that we're waiting longer than ever to get the life-saving diagnosis that we need. And that by the time clinicians review the scans, they're spending less time than ever, opening up the possibility of misdiagnosis. So this is why Harrison AI exists. We built medical-specific AI system that can analyze case, provide diagnosis and descriptions, alerting care team of cases that need urgent treatment, and of course, producing draft reports, documenting findings, saving clinicians time. This AI system allows for accurate diagnosis at the moment that matter, and to scale our capacity in looking after patients that is currently not possible with human clinicians alone. Let me be clear, this is a very hard problem. One of the challenges is that modern AI systems that many of us have in our pocket today, like OpenAI, Anthropic's, Gemini from Google, simply was not built for this purpose. They've been trained on large corpus of internet data, but unfortunately, the internet do not contain sufficient quantity and quality of medical imaging data to train this system with the expertise it needs to make critical diagnosis. But more fundamentally, the very task of providing a good answer is not the same as making the correct diagnosis. This is where Harrison is different. We train our AI system on large corpus of identify and anonymize medical imaging data and fine-tune that with radiologists, clinicians in the loop, providing reinforcement learning feedback with the sole goal of providing accurate diagnosis for patients. And the number shows that. Harrison AI recently was tested against the FRCR Rapid 2B exam. One of the neat things about medicine is their credential exam for everything. This test, in particular, was designed by the Royal College of Radiologists. Every radiologist, before being credentialed to practice medicine, need to pass this test. It's a test that scores out of 60, you need to score over 50 to pass. It's very simple. 100 case radiologists need to be making a decision about normal or abnormal and make the right diagnosis very quickly. As you can see here, the radiologist barely passed this. At 50.64 out of 60. Most modern LLM systems today score barely above random guessing. This is not so surprising. I'm sure many of us has tried to put our own X-ray on some of these chatbots before and really got mixed results. You can see here Harrison AI recently passed the threshold of clearing the passing score for this credentialing exam, and of course outperforming the average score of radiologists. So we built radiologists as an API through this AI system. Now, the other really interesting way of evaluating the system is through what we call a Turing test. We've all heard about the Turing test before, but this one was designed by Mass General Brigham University Hospital and Harvard and the American College of Radiology. The test generate thousands of radiology reports using AI and put that against thousands of reports generated by radiologists on a blinded fashions, and ask 113 radiologists if they can tell the difference and whether or not those reports they would feel comfortable signing off. And as you can see, we have some way to go, but the AI is accepted by radiologists 65% of the time. That doesn't sound like a lot until you realize the radiologists themselves only agree with their peer 79% of the time. So we're getting incredibly close to that human level performance of expert radiologists. The reason why this is so exciting as a technology is because of the benefit of software scale. Once we built the technology, it can never be unbuilt. And it can be scaled infinitely through our cloud infrastructure, anywhere, anytime, up or down, absorbing the capacity requirements of diagnostic need that are growing all around the world. Today, Harrison AI system is being deployed across 40 different countries, impacting 10 million patients' lives annually. We're processing 35% of all chest X-ray in NHS England. Every CT brain in the public department, emergency department in Hong Kong. And here in Australia, 50% of all radiologists are using at least one of our tools on a daily basis. This would make us the most scalable radiology department ever built. And that is the power of software and AI in healthcare. I'll come back to this. Ultimately, it is all about the human story. Patients like Diane can breathe easier because we have the power of technology that can process the case and provide a critical insight in moments that matter. About Harrison AI, we started the company just over 5 years ago, right within the dorm room in University of New South Wales here. At the time, Dimitri, my co-founder, and I sat down and set ourselves an ambitious mission: 1 million daily recurring live touch. That's 1 million scan or million cases processed through our system every single day. Just 2 years ago, we blew past the 1 million lives per year, and recently, as a company, we celebrate our milestone of 10 million lives impacted per year. Now, if you do the math, we still have a long way to go to achieve this mission, and we'll work hard at that. AI will change the world in many ways across practically every industry. But this one of healthcare is worth solving and worth solving right. Thank you so much.
Thanks so much, Angus. It's hard to believe you were only founded 5 years ago and now impacting 10 million lives. That's truly incredible. Our next speaker is Dr. Sebastian Himbert from SynthMed Biotech Incorporated. He's the co-founder and chief technology officer in SynthMed Biotechnology, which is a Canadian startup pioneering smart drug delivery systems using engineered red blood cells. He holds a PhD in membrane biophysics from McMaster University, where he continues as a postdoctoral research fellow. Sebastian's research focuses on manipulating cell membranes to overcome the barriers such as the blood-brain barrier, enabling targeted therapies for neurological and infectious diseases. With over 20 publications and several patents, he combines deep scientific expertise with entrepreneurial vision to advance precision medicine and nanotechnology for global health. Welcome, Sebastian.
Yeah, good afternoon, everybody. And it is really a privilege and pleasure to be here this afternoon and share a little bit of the journey that I had from doing a PhD in biophysics to eventually becoming an entrepreneur and developing new red blood cell-based therapies. Now, I want to start with a little bit of my background story because you may hear it on my accent, but I'm neither from Canada nor from Australia. I'm actually from a small town in southwest Germany, right at the French-German border. And this area is known for a beautiful landscape, excellent food, but most importantly, it had a biophysics program that I enrolled into, and we were only like 20 students. And this kind of close environment made it ideal to kind of learn with close interactions from the professors, all the details of biophysics. But when you live in such a small, tiny village, you actually want to explore the world a little bit further relatively quickly. So I decided to move across the Atlantic and ended up in Toronto, where I eventually completed my PhD in 2021. And this PhD was focusing on particle physics. Especially, I used particle physics as a tool of microscopy in order to study the molecular and structures of red blood cell membranes. Now, you may all be familiar with traditional light microscopes, where you can see here on the left-hand side the microscope picture of cells. But with a light microscope, you cannot look smaller and smaller. In order to see tinier structures, you actually need to use particles in order to resolve proteins and membranes and other molecular pictures. In my PhD, I used particle accelerators in order to study the structure of red blood cell membranes. And the idea is pretty simple. You take a particle accelerator or a nuclear reactor, you shoot the particles at the material you want to study, and what we as a physicists do, we actually look at how these particles disperse from the material in order to understand how they look on the molecular level. And for the first time, I was able to measure a molecular high-resolution image of a red blood cell membrane. And this gave me insight into all the details of how these membranes look like, how they move, how they organize themselves. And this was, at the time, the most detailed image of a red blood cell membrane that we had, and which became eventually critical to the work of what we're doing at SynthMed Biotechnology now. Doing research on blood requires, first of all, a lot of blood. And then, well, if you're wondering where all this blood is coming from, well, I was quite literally bleeding for my PhD. And if you're wondering how this sounds like, I don't know if the audio was good. Anyways, they missed the veins quite often, and I had a number of blood.
Shit.
So, aside from bleeding for my PhD, I'm also quite passionate about physics. I'm passionate about programming, biochemistry, tinkering, and creativity. And all of these kind of skills, interests, and multiple disciplines is what brings us together at SynthMed Biotechnology, where we actually innovate the world of drug delivery. Now, if you take a pill, say Tylenol or Advil, for instance, the drug actually distributes itself all across your body. Now, for ibuprofen, that's not a problem. But for many drugs, you actually want the drug to go to the target of where it is needed. You want to have it ideally in your brain, in your lung, depending on where the cure is needed. And for many drugs that are currently in the clinics, a lot of them actually fail because they have a short half-life in the circulation, they have a high off-target activity, or they have a high dosing requirement. And this eventually lets a lot of drugs fail in the last phases of the clinical trials and makes something that is actually potent unusable. So in an ideal world, we want to bring this drug just to where they where they are needed, such that they can fulfill the therapeutic purpose, say in your brain, but leave the rest of your body unattached. And our vision is to use red blood cells to bring the drugs to unreachable corners of your human body. Red blood cells are the natural transport mechanism that all of us have. They transport oxygen, nutrients, and they are highly biocompatible, non-immunogenic, and have a long circulation half-time. So ideally, those are pretty much the ideal transport mechanism that your body creates. But unfortunately, the red blood cells on their own are not specific enough to bring any drug to the target that you want to deliver it to. So we need to outsmart the system in order to get the red blood cells being useful and being functional. And now comes into place what I did on my PhD. We studied the membrane structure on a molecular level, which means we can actually also tune and manipulate the membrane of these red blood cells on really the tiniest levels possible. What we developed at SynthMed are nanocarriers, about 1,000 times smaller than a human hair. They use the membrane of red blood cells, but we dope the membrane with additional synthetic compounds that eventually enable us to load the therapeutic compound inside these nanocarriers and put a targeting moiety on the surface. And both of this allows us to eventually bring a therapeutic compound to the target that we want to deliver it to. We were talking about how long such an innovation actually takes, and I can tell you that I started my PhD more than 10 years ago, and this is about the time that it took in order to get to where we are today. We had some remarkable successes along the way. We developed the COVID vaccine during the pandemic that was based on red blood cells and delivered antibiotics successfully to bacteria. But at SynthMed, we are focusing nowadays on two major challenges: on delivering drugs to the brain and to improve the outcome for cancer patients. The brain is one of your most precious organs. As such, it is highly protected by a barrier called the blood-brain barrier. You get oxygen through it, you get carbon dioxide through it, and also the good stuff like alcohol and caffeine. But fortunately, it protects pathogens from reaching the brain. And then on the unfortunate side, it also protects many drugs from reaching the brain. And overcoming this challenge is one of the most difficult ones in modern drug development. Now, we have developed carriers using our technology that actually can penetrate this blood-brain barrier. By using our red blood cell-based therapeutics, they circulate in the body. You just do an IV injection and eventually bring the drug into the brain. The drug that we are loading into our carriers is a memory-active compound. Now, it is an endogenous protein that is present in all of your brains right now. And as your memory forms, when you're listening to my presentation, it actually works on forming the synapses in your brain. Now, if you have Alzheimer's disease, the concentration of this compound is lowered in your brain. And the idea that we initially had was very simple. We just bring additional amount of this protein back into your brain and hopefully can offset this memory loss. We are fortunate of working together with one of the leading scientists in the world on this particular protein that we are using. And with her advice, we were able to run preclinical tests on animal models. I'm just showing you how we actually test memory in animals. A mouse is not the smartest animal around, but we can test their memory by putting them in a pool. See here, like a water pool, and a mouse is placed into the water and then finds this island that is here like circled in the red dot. The mouse itself doesn't see the island, but it sees this black dot that we put on it. And the mouse relatively quickly identifies this is a safe spot to go and eventually finds the island within a couple of seconds. In this process, the mouse actually memorizes how to get to this island. So then one day later, we remove this marker from the island. The mouse can no longer see where to go and how to get to this island. But it needs to rely on its memory to find this island and the safe spot in the pool. Now, if you have, I think some of the videos got mixed up actually in the slides. So what you see here is a mouse that has Alzheimer's disease. And this mouse actually takes about 120 minutes in order to circle in the pool, and it never finds the island in this time. If you run the same experiments with a healthy mouse in comparison, you can see within just a few seconds, this mouse remembers finding this island without any difficulty. Now let's look again at a mouse that has Alzheimer's disease, but has been treated with our technology. Again, reminder, we bring this memory controlling compound back into the brain of the mouse. And within just a few seconds, this mouse has no problem finding this island again. And we have tested this with more, thank you. We have tested this with a large cohort of animals, male as well as females, and throughout the trial, we actually see a significant improvement of the memory function of all the treated Alzheimer's diseased animals. So the shorter the time, the better the memory of the mouse is. Showing you how we get how we can improve the memory in animals is just one example of how our technology can change how therapeutics work. As I mentioned, the other goal that we achieve at SynthMed is changing the outcome for cancer patients. In the case of cancer, there are many ways of treating such devastating disease, but the most recent approaches focus on an immune therapy approach. Here you deliver the antigen of the tumor to the immune system. You trigger the immune response in order to promote the immune cells to kill the tumor cells. This idea is not particularly new, but it has been tried for several years now. The most simplest approach is you take the protein, add an adjuvant to it, and treat the patient with a combination of both the adjuvant and the protein. We have done tests in animals to test competitive technologies and our technology. We do this by taking a mouse again, we transplant the tumor into the mouse and then let the tumor grow. And what you see here on the right-hand side is the size of the tumor over the time. You see, if you have like a very basic approach, the tumor grows relatively quickly. Within 10-15 days, the tumor is so big that the mouse is at the end of its lifetime. Now, let's look at how the state-of-the-art technology works. The LNP mRNA technology. And all of you who went through the pandemic remembers this kind of technology. This was literally the COVID vaccine. Now, it works a bit better, but fair enough, after 20 days, the tumor grows exponentially and the mouse still doesn't survive. With our technology, we can actually expand this suppression of the tumor past the 30 days, and we have even data that go up to 50 days without, by suppressing the tumors successfully in all the animals. You can see how, while it doesn't completely remove the tumor, this technology can actually improve the therapeutic chances for any cancer patients. The two examples, what we are focusing on at SynthMed, are just examples of how the technology can be applicable in the future. In fact, we have developed a system, as Solina likes to call it, the FedEx of drugs, where we can bring drugs of any kind of format or size to the right target in your body. our vision for the patient experience is that the patient comes into the clinic, we take about 10 milliliters of blood. That's a blood collection that everybody does regularly for routine checkups. And this allows us to synthesize enough dosages within a day of a process before the blood eventually gets injected back into the patient. Now, I hope I was able to show you a little bit of how I got from a PhD to what we're doing at Synsmed and got you excited about our technology. But I wanted to end with just saying that I was bleeding for my PhD, but I'm still bleeding to make this red blood cell therapeutics work in the future. Thank you very much.
Thanks, Sebastian. There are many of us in this room who feel like we bled for our PhD, but you have taken that to a whole new level. Very impressive results in that mouse model. Our next speaker is Dr. Andrea Cho, who is the CEO and co-founder of HoloClara, the first biotechnology company to create safe, orally available worm-derived therapies designed to provide relief to millions of people living with allergic and autoimmune disease. The company's investors include Bold Capital Partners, Horizons Ventures, TerraEsk, Endurance 28 and Free Flow Ventures. Dr. Cho received her MD at USC Keck University of Medicine and her PhD at Caltech. Her doctoral work was on the evolution of a symbiont chemistry, which laid the foundation for the technology at HoloClara. She founded HoloClara with Paul W. Sternberg, PhD, professor at California University of Technology, and Frank C. Schroeder, PhD, professor at Cornell University. The company is currently valued at around US $80 million. Thank you so much, Andrea. Welcome.
All right. Is this thing on? Yeah. Okay, well, my name is Andrea, and I'm very excited to be here talking with all of you. Trying to find the right clicker. Let's see. I'm going to try again. Sorry, I don't know. It seems to not be working. Oh, there we go. All right. Okay, well, before I start talking, how many of you are actually thinking, what the heck does HoloClara even mean, and what is this lady going to tell me about worms? Right? I usually never start talks talking about the name of our company, but I thought actually today that I might. HoloClara actually comes from this word holokleros, which means to complete. And I want to invite you to think about what you need to complete your human experience. And I'm not talking about this season's Louis Vuitton bag, although I wouldn't turn that down. I'm talking about in your more quiet moments, like when you're by yourself or when you're with your loved ones. Has there ever been something that's held you back from really enjoying a simple moment just because someone, either yourself or a loved one, is in pain or disability? And just to paint that picture, let's envision you wake up in the morning and you think, I want to have a perfect flaky chocolate croissant with a fresh squeezed orange juice and a perfect shot of espresso, and I want to listen to my favorite song and not have any worries, right? For some of you, that might be Taylor Swift's new album. Any millennials? Joshua Tree, you too? Come on. You know, for me, it might be Mozart, Marriage of Figaro. I don't know how many of you know that. Duettino, one of the most beautiful songs you'll ever hear, of two voices soaring in the air. Now imagine not being able to enjoy that moment because either you have to think, oh my gosh, I need to find a bathroom after this. Oh, I have to work. I actually need to figure out excuses to tell my coworkers because I don't want them to know that I have a disability because they may actually think that I'm weak. Or maybe you cannot enjoy that moment because a loved one is suffering near you and they have bed sores, they're bedridden, they're being turned in a way where, you know, parts of their body that no one ever really wants to share with someone else is now visible to a stranger. So those are the types of moments that I'm talking about, that it is my mission to try to complete that experience by trying to find things out there that can complete our human biology. And that's what our mission is at Holoclara. So here's a picture of Earth. Look at how beautiful it is, majestic Earth. When I think about this image, I think about what makes it so beautiful, and to me, it's probably nature and people are what make it beautiful. And I hope that by the end of this talk, I'll inspire you to wonder about what are the nooks and crannies of this planet that we can find new medicine for our people, and that in exchange, maybe we'll take good care of the Earth. And so of all of the people that live on this planet, millions are actually still suffering despite modern advancements. Does anyone know this artist? Say it out if you know it. Paul Klee. He actually died a very long time ago and had scleroderma, and in his later years drew this painting to demonstrate how he felt like his body was falling apart. He died a very long time ago, and yet we still do not have a cure for scleroderma. We have such incredible modern advancements, like a ring that'll tell you how you're sleeping. We have AI that'll tell you if you have a tumor in your lungs. Now we have targeted, ultrasound to dissolve that tumor. I mean, there's all of these incredible technologies. Genetic disorders, where now the hope of CRISPR can help that. But what about chronic inflammatory disorders? We still don't really have a great cure for that. And in order to really find something, we need something new. I'm not talking about iterating an existing drug to get a piece of the market. I'm talking about something completely new. And if we really study the history of breakthroughs, you'll see that actually a lot of them came from nature, including CRISPR came from bacteria. Did you know that? If you ever watch the Netflix documentary, you'll see that actually yogurt scientists were involved in this discovery while they were studying yogurt bacteria. These technologies, penicillin, statins, all of these came from nature and have helped millions of people. Botox has helped millions of faces, right? Ivermectin, Taxol, Ozempic. Did you know that actually came originally from the secretions of a Gila monster saliva, right? And so these are the places where I'm talking about opening up a whole new class of drugs. And where would we find yet another place that we can look for new medicine? Well, here's a clue. A big clue I'll tell you is tiny creatures. And today I'll talk to you about how worms can inspire the next generation of med tech. Why worms? Well, you know what? To be honest, I don't actually really know. All I know is that there's a lot of proven science. There are really reputable institutes and really reputable journals that are publishing that worms, for some reason, can improve lifespan, fertility, metabolic health, inflammation, obese mouse, non-obese mouse, fetal stubs turning on. It's really incredible what they can do. So the question is, the million-dollar question is, well, what are they doing? How are they doing it? And before I got my medical degree, I actually got my PhD at Caltech decoding the language of worms. And there, with many brilliant scientists, we together characterized the first Rosetta Stone of the worm language. And so people always ask me, well, how do you get from there to medicine? Like, what's your secret sauce? I'm like, you know what? I'm going to create a slide today on the secret sauce. A simple way to describe it is, once you know what those worms are saying in molecules, you can actually put them into one capsule, let's just say, and then you can separate out that language, and you test them individually, you give it to sick mice, and you see which one makes it better, right? So then from there, you go from the quiet observation of a laboratory into the hustle and bustle of a startup and a company surrounded by incredibly brilliant people that are chemists, patent experts, pharmacologists, regulatory agents, and clinicians that help take nature's innovations into tomorrow's medicine. So I'm very excited to share that our first worm molecule actually went to trial this year. And we successfully completed our first clinical trial which, by the way, for any MD, PhD, it is our dream to go from benchtop to bedside. So, any guesses where this trial was done? Quiet audience today. Come on, someone yell out a country. Australia, yes. Go Australia. Did you know that you guys are actually global leaders in clinical development? I mean, it was really remarkable. We had 56 Australians enroll in our trial and take this medicine. And with incredible safety, we can now turn curiosity into care. And from here, we will now work towards going to patients, figuring out how we can help all of these people suffering from asthma, right? There's some of you out there that carry an EpiPen, right? Do you guys know what that is? It's kind of like, didn't Uma Thurman have that in Pulp Fiction? It's like literally a vial of adrenaline being stabbed into you if you can't breathe. And then people with inflammatory bowel disease, which actually that's the story that I shared with you earlier about someone that can't really enjoy that croissant. And then eosinophilic esophagitis, which doesn't just affect adults, but it also affects children that can't swallow food. They can't eat food, they cannot swallow food. And so, we're really excited to move towards these patients and ultimately serve them so that they can live more complete lives. Okay, enough about us. Who wants to hear some cool science? All right, that was a trick question because I'm still going to talk about us. But I'll just start by saying we know that there's other cool worms out there, right? Like I showed you all of these incredible journals, metabolic health, longevity, fertility. How do we go after them, right? So there's always been this question in my mind, all right, like our first molecule's up and running, it's in clinic, it's doing great, but there's other molecules from worms and I want to get them, so how do we get them? Well, we do what we also did, and I showed you, we divide up the language and we test them individually to see which one works. But are there other cool ways to get them? And I'm going to tell you a story, actually, because, well, that's how I stumbled upon this technology, so I thought maybe I'll just share with you how I discovered it. I was walking into Caltech professor's office, and he's a brilliant synthetic chemist. It was actually with his help that we were able to make 1 kilo of our first worm crystal. He's running late. It's like 5-10 minutes late. No big deal. And he goes, "Oh my gosh, Andrea, I'm so sorry I'm late. Something crazy happened today." I'm like, What? Tell me. He goes, "We uploaded a paper and it went viral." And I'm like, okay, first of all, that is the ultimate badass move, is when you're a professor and you no longer care about getting into a public, like a peer-reviewed journal, and you're just uploading your data. And he said, "Well, yeah, it's just remarkable." I'm like, "What? Tell me what was the paper about? I want to know." He says, "Well, Andrea, did you know that actually in analytical chemistry, there has not been a revolution in the last century? I mean, more or less, we have improved mass spec and chromatography and NMR, but we still use this." I'm like, come to think of it, when I go into these labs, this stuff does look monolithic. And he says, "Well, and that, people won the Nobel Prize in structural biology by using cryo-EM to elucidate these proteins. No one's ever thought to try to use them in small molecules," which, by the way, the worm language, small molecules. And he said, "Until now." I'm like, "Okay, it was you, right?" And he says, "Yes, it was, but also with a colleague of mine at Caltech." And here is actually his reflection. You can see that this is him sitting in front of the Talos Arctica, which is a multi-million-dollar machine. Costs about actually $500,000 a year just to maintain. And it can shoot lasers faster than the speed of light at a sample and determine atomic resolution instantly. So this is not their paper because, like I said, they just uploaded it to the internet. But everyone wrote about it. This is a Nature review where they talk about how these Caltech scientists prove cryo-EM can elucidate structures in atomic resolution in minutes. Here's an actual quote from the paper. People are smacking their heads, saying, "Why didn't we do this earlier?" So my first thought is, can you put worms under the laser, right? Can we do cryo-EM on worms? And the answer was, I don't know. no one's ever tried, which is my favorite response because my next question is, can we get a discount? And that's exactly what we're doing now. And I'll show you, this was actually from a couple weeks ago. You see by my nail polish color. That's me handing over some samples of worm bodies and heads. And then just recently, oh, I love this obligatory Caltech scientist Red Bull in the picture. We were able to make crystals from our worm samples. So for Christmas this year, we will be seeing what's coming out of these crystals. And for me, that's the carrot out at the end of the stick, because more worms can help more people. Truly, that's our goal. And before I wrap, I'll say this is the last chapter of my talk, and the shortest, don't worry, it's almost over. I'm sure some of you have thought, but why? Why do worms make medicine, right? And it could be partially due to the fact that for some reason, areas of the world that still have gut roundworms are spared from these inflammatory diseases. Far less type 1 diabetes, rheumatoid arthritis, multiple sclerosis, inflammatory bowel disease. There's something about these gut roundworms that are protective. Could it be because we actually have had them for a long time? They are our old friends. They've been with humans for millions of years as human ancestors have evolved, making this medicine to benefit their host. And is it a coincidence that when we abruptly remove them because we thought they were so gross, that suddenly there was a historic surge in all of these diseases? Well, I can't answer that question, but I invite you to consider that possibility. And so, when you now look at this world, I want you to think about the bright future ahead, all of the millions of people enjoying their croissants, you enjoying, walking with your best friend, having a laugh, being able to enjoy beautiful, quiet moments, both on yourself and with your loved ones. And I invite you to join me on this journey of keeping hope alive, helping these people just to not only enjoy their moments, but to live complete lives. Thank you.
Thank you, Andrea. We all have a new appreciation for worms, and we'll be signing up for worm language classes, no doubt. Truly incredible work, and as someone with Crohn's and celiac in my family, I'm just so excited to see you bring that incredible science into the real world. We now have a break, and there's time to check out our MedTech Expo. So go out and have a look, have a coffee, make new friends, talk to new people, explore new technologies. We will be reconvening at 4:02 precisely, so you won't forget that, 2 minutes past 4. And I want to just encourage you, after the break, we have Senator Dave Sharma joining us, but we also have the histosonics demonstration. Now, I've known Bill Barty for quite a while from my time at CSIRO, and if he's looking forward to that, then I am beside myself with excitement. Do not miss it. So see you back at 4:02. Welcome back, everyone. The veil has been lifted. This is the moment we've all been waiting for. So Histosonics is a pioneering medical technology company developing and commercialising histotripsy, the world's first non-invasive platform that destroys, liquefies and eliminates tumours and diseased tissues without incisions. So Dr. Zhen Zhu is a Li King Xing Endowed Professor of Biomedical Engineering and Professor of Radiology and Neurosurgery at the University of Michigan. A pioneer and world leader in histotripsy, a groundbreaking ultrasound therapy that mechanically destroys unwanted tissue, she co-invented the technology and co-founded Histosonics, which is commercialising histotripsy for non-invasive cancer treatment. Her research is advancing applications in cancer, neurological and cardiovascular care, leading to the FDA approval of the Edison system for liver tumour treatment. Dr. Zhu is a Fellow of the National Academy of Investors, AIMB and IEEE, has served in leadership roles with IEEE and ISTU. Her contributions have earned her numerous accolades, including the IEEE Karl Helmuth Hertz Ultrasonics Awards in 2024 and the Lockhart Memorial Prize for Cancer Research. Please join me in welcoming Dr. Zhu.
Thank you, Professor Fox, for the introduction. All right. Well, thank you, everyone, and it's really my great pleasure to have the opportunity to talk about histotripsy and histosonics. So what is histotripsy? The term, when we actually coined histotripsy in 2002, because all medical terms is based in Greek, so histo means soft tissue, and the tripsy means mechanical breakdown in Greek. So the intention is that it's the first non-invasive technology to perform the real surgery, meaning that do it in a mechanical way. So imagine that you have ultrasound delivered from outside the body. See, this is the ultrasound device here, and it's focused in the target tissue with a millimeter accuracy. So what you're seeing here is you are seeing a cluster, a cloud of microbubbles generated, and it's about the size of 2 millimeter by 4 millimeter also, and it's 14 centimeter away from the ultrasound transducer. And that bubbles kill the target tumor without incision, without radiation, without injury. And that's what I'm about to show you. So what are the clinical benefits of histotripsy? Why do we need to do it? First, non-invasive. As I mentioned, that the ultrasound is actually delivered from outside the body. And what you are seeing here is an actual patient case. There's a tumor actually deep in the liver. If this patient is treated with conventional surgery, what you will see is a lot of injury to the liver here. But this patient was treated with histotripsy. And histotripsy liquefied that tumor to acellular debris. And that acellular debris is absorbed over time by the body. And you can see 30 days later, that tumor is actually not tumor anymore. It's just liquefied debris here. And it's substantially reduced already. And most importantly, there's no damage to the surrounding or overlying liver at all. Second, high precision. So what we are doing here is we rely on using ultrasound to generate microbubbles, which is termed acoustic cavitation. And that is a threshold phenomenon. So bubbles are only generated when the focal ultrasound pressure is above a threshold. You can see actually the histology slide of tissue treated by histotripsy. And typically what you see is you have intact cells like this. In the histotripsy treated region, the cells mechanically disrupted entirely. Okay? So those are debris, then the body just cleaned it up. But what you will see here is you can see at the boundary, there is a half of the cell, and the half of the cell cut off. It's because our effective scalpel here are microbubbles on the size order of a cell, and incredibly precise. If you have pressure above the threshold, you have damage within the treatment region, and if it's below the threshold, then you have no damage at all. Therefore, we don't see the same partially damaged region like radiation you will have seen, or thermoablation then caused by actually heat perfusion. So it's a highly precise procedure. Third is real-time image guidance. For a non-invasive procedure, one thing is that the clinician is not seeing everything with their eyes directly because they're not opening the patient up. However, with histotripsy, what we are doing is we have an ultrasound image. And what you are seeing here is from a patient treatment, and the red circle here is the tumor outlined by the clinician that he's trying to target. And then the red cross here indicates the ultrasound focus. And see the bright spot here behind the red cross? And that is a bubble cloud, a cloud of bubbles generated by histotripsy. And the bubbles are natural contrast agent for ultrasound. Therefore, you can see the bubbles generated in real time. And that help the clinician to guide and monitor the treatment, even without opening the patient up, to maintain the high safety and the treatment accuracy. Next, most of the patients that they wake up from histotripsy procedure, by the way, it's a one-session treatment. The patient come in, and the clinicians put them down and just scan and remove the tumor, and that within depending on the size of the tumor, within a couple of hours they wake up, and our record right now is actually 10 tumors in one session treated. But typically, patients wake up, the first thing they ask is, have you done anything to me? Because they don't feel the pain. There's no pain. They immediately should go home and have dinner at home and being able to return to normal activity the second day. So we're talking about a form of cancer care, not like chemo or radiation therapy has significant side effects. It's non-invasive and also increase the quality of life. You are getting a tumor treatment with dignity and allow you to still live a normal life. Next, tissue selective. So what we found out is that the critical structures in our body, such as normal blood vessels, and this is actually, by the way, is a histology showing liver tissue treated by histotripsy. So you can see the critical structures like normal large blood vessels and nerves and the bile ducts. They actually remain intact in the histotripsy treated region when the surrounding liver tissue or tumor tissue get completely destroyed. It's because those critical structures, they naturally have a high mechanical strength and more resistant to the mechanical damage. And this allows us to actually go in, and we have seen in treating high-risk locations. We have seen clinicians treating a tumor sandwiched between vessels that is surgically inoperable. And the clinician was able to go in, destroy that tumor, and the vessels remain entirely intact. Lastly, was a surprising finding from our research, which is this immune stimulation. And I will talk about the mechanism more later. So it turns out that histotripsy also stimulates this potent immune response, and such that in selected patients, what we have seen that is treating one tumor can lead to substantial regression of other off-target tumors. And this is in a patient where this patient really coming very late stage, liver look like Swiss cheese with so many tumors in there, and the treated tumor is not even on this image slice. But what you see here is, look at all those tumors. And 8 weeks later, substantially regressed due to the immune stimulation effect induced by histotripsy. So I'm going to talk about 25 years journey to bring histotripsy from a concept And to the first time, I observed the phenomenon as a PhD student. And eventually, this whole map. So 2001, I started my PhD, and 2002, first observed the phenomenon. And 2004, I published the first paper on histotripsy, and we coined the term histotripsy at University of Michigan, and I was in that publication. And spent the almost 10 years to really trying to design and build a specialized equipment for histotripsy and understand the mechanism behind it. And December 2009, co-founded Histosonics with a goal to really bring this to the patients. And 2016 was the first clinical trial, actually, and it was performed in prostate, so benign prostatic hypoplasia. So that was the first human trial, and unfortunately, the results from that clinical trial was mixed and didn't meet the goal. And years later, we found out it's because the human patients and our preclinical animal model, they don't actually match well. We switched to liver, and 2017 built the first liver human system, and 2019 had the first human trial in cancer and also in the liver. That was in Spain, and the results were fabulous. And that led to 2021, the multi-center trial we call Hope for Liver trial. And guess what? COVID. So the trial was really hard to do, as you can imagine, from recruit patients, the whole thing. But however, the results, again, super positive. And that led to the FDA approval in 2023. And it's for non-invasive histotripsy treatment of liver tumors. And 2024, we started a clinical trial for Hope for Kidney, so for treatment of renal tumors, because it's a platform technology. So it really can be used to treat any solid target. Again, going really well, enrollment complete, and hopefully the data will be submitted to FDA soon. And we started in December 2024, the pancreatic cancer trial. And it's ongoing, and the results look really good so far. And I was told the number, I showed 25 patients treated, but the new number is, I think, 3,000, actually over 3,000 now. So we have treated over 3,000 patients in the United States, in Europe, UAE, and Asia, around the world, and now hopefully in Australia soon. And 2025, really recently, had this recent acquisition, now evaluated the company at $3 billion. So 25 years in one slide. And I was trying to break down to the key critical moment early on before you can actually see the demo. So 2001, why actually at that moment that inspired me to look into invent histotripsy? So I remember I was a new PhD student at University of Michigan, and there is this pediatric cardiologist. His name is Ahi Ludomirsky. So he came in and he asked one question. So he was operating on pediatric patients, very sick patients with congenital heart disease. They are very little kids and babies, and they were dying on his operation table because they were so sick that they couldn't endure the invasive surgery. So he came in, he asked our research group at biomedical engineering department, he said, can you invent a non-invasive interventional technology that can perform the real surgery? And he emphasized on the real surgery. And why? Because there was already, there's radiation, right? And there's a different kind of form of thermal ablation using different energy to generate heat to kill the tissue. But they leave the tissue in situ. Even you kill the tissue, you're not removing them, not like a real surgery. And he needed something to do the real surgery for his patients with congenital heart disease. So he asked, can you actually invent a non-invasive way to perform the real surgery to allow me to save those really sick kids? And I was in the audience, and I thought, this is such a wonderfully impossible idea that it's a perfect PhD dissertation for me. So I went to Ahi and went to my advisor and said that I would love to take this as my PhD dissertation. And my mentor looked at me for a second, and he said, well, if you do this, my advice is do not read any papers for the initial time, because I do not want your innovative mind to be restricted by conventional thinking, which he means that the current literature says is impossible. Okay, but I started it. So my first year of PhD life involves I go to slaughterhouse every week to do dumpster diving of pig hearts. Now I bring to the lab. Literally, when I say dumpster diving, I'm not even kidding, because I literally go in and they just give me a black trash bag. And then they showed me where a big trash can is, full of pig organs. And then they charged me $5, but I can fill the trash bag with whatever organ I want. So that's what I meant. And no gloves, right? You go in and you get it. So anyway, so I did that for for the first year. I go in, I get pig heart, and I come back and I dissect them, put them in the water tank. So what you'll see here is a piece of pig heart tissue in my holder, and I have ultrasound transducer that placed actually 10 centimeter away, and I trying to target. And then I was using all kinds of parameters and trying to test. And as expected for the first year, it was failures after failures and after failures until this day. And this is the experiment that I did, and you are here to actually see what I saw in 2002, more than 20 years ago, that first experiment. So what you're going to see here is that I want you to actually look at this spot. And this is in a water tank, by the way, okay? So you will see a video here, and the video here is I run over to turn on the amplifier, and you will start seeing almost like a smoke coming out. But remember, it's not smoke, because it's a water tank. You can't get smoke at all. And this is all in real time. So you see kind of smoke coming out, then you start seeing a minute later a beautiful perforation through the pig atrial wall tissue. So what happens here is that was the first time I witnessed that using ultrasound with microseconds length pulses at really high pressure that allow me to actually break up tissue to sub-micron size ACL debris. That's why it looked like smoke. And again, like I said, a minute later, I see a perforation. Sound is not touching here, okay? So I can tell you that I did it first time, and I thought I was dreaming, because after a year of failure, I was like, this is probably just me trying to, wish this will happen. So I did it second time. And I still couldn't convince myself this is real. And my rule of thumb is if you can repeat the same thing 3 times, it must be real. So I did it 3 times, and I remember I need to turn on the camera. And did it 3 times. And again, it happened again. And I ran over to Charles Kane, my PhD mentor's office. I said, Charles, you need to come over here, because something very special just happened. So that was the first time I actually saw histotripsy. However, after that, within about a month or so, and I want to actually, before that, I should show that this is the equipment I actually used back then to generate the first histotripsy experiment. You can see this is a transducer from 2002, and that was a single element 788 kilohertz transducer, and it's run by this amplifier. So it's one element and run by this amplifier. This amplifier was the most powerful amplifier in the world at that moment, and it's ENR E1000 amplifier that gave you 1,000 watts of the power to drive the transducer, and it has to be run by a 380 volts electrical power, right? We really have only one plug in the entire university that can enable this amplifier to run, and the cable is 4 feet long, and I claim that space. Like, nobody touch it. This is my space. I'm going to cover this with pig heart. You don't want to touch it anyway. So that was 2002. But however, a month after my first successful experiment, I had to run over to Charles' office again. And I said, Charles, I have good news and bad news. The good news is I was able to repeat the experiment. So this is real. The bad news is that I just broke the most expensive equipment in our entire lab. But I don't think it's my fault. I think because we don't actually have a histotripsy work back then, I said to make this work, even the most powerful equipment, commercial equipment at the time, is not sufficient. If we are serious about this, we have to design and build our own specialized equipment. Should we do it? And Charles, as crazy as I was, said yes. And it was funny because we knew that we couldn't do it with a commercial equipment. So when we published a paper in 2004, the first paper, the entire scientific community, when after reading that peer-reviewed paper, had one response, and the same response that they kept telling me for 10 years, which is, it's impossible. Histotripsy is impossible for two reasons. One is you would not be able to generate the histotripsy parameters in a robust way, even with the best commercial device. And second is we don't know how it works. Mechanism is entirely unknown, and cavitation, which is microbulb activity, was a phenomenon have known at the time to be entirely random. For 50 years, nobody can control it, so you can control it either. So that was the thing, the kind of community response. So first, we spent 10 years almost to trying to design and build the specialized device. So I just want to fast forward 20 years later. Don't know why it's blurry, but 20 years later, this is a more recent device we built. So this is a single element transducer. Here is a 260 element histotripsy ultrasound array. So 260 elements, which we build by 3D printing. And this is an amplifier. Remember I said this is an amplifier that's as tall as me, right? And drive one single element. This is an amplifier, has 8 channels. It can fit on my palm. Each one has 4 times power than this. And not only it can generate histotripsy, it can also receive signal to do imaging as well, to help us with monitoring and doing some really fancy stuff. And if you actually, and so now people don't think it's impossible anymore with this device, but it took us almost 10 years to even get started and 20 years to really get to this form. Second, about the mechanism. And if one thing is unknown for a scientist, it's opportunity. So what we did is I set out and we went and we got ultra high-speed camera and a microscope, and we started studying bubbles, how bubbles work. So what you are seeing here is the 2 critical things of the mechanism. The first thing is how microbubbles can be generated in a controllable and predictable fashion with ultrasound. So what happens is that we are breathing air, so we have nanometer gas pockets in our tissue because it's filtered by the lung. So it's wonderfully stable. The surface tension is actually 200 times of the atmosphere, really high. But however, imagine if you have a gas pocket, like a balloon almost, and you can make the pressure outside the balloon lower than the inside, which means physics-wise, overcome the surface tension of that gas pocket. You can make it actually expand, and eventually it will collapse. Which means that if we can, within microseconds, generate over 200 times of the atmosphere, but negative pressure, because remember, I need to go the pressure outside than the inside of the balloon, so negative pressure. Then we can actually expand the gas pockets and later collapse. So what we happen to see here is Charles King, my PhD advisor, Adam, Eli, my former PhD students, we were a team. Now they're professors too. But we spent almost, I would say, 15 years in doing some crazy experiments together. So what we show here is that, if you're actually looking at tissue, most tissue are water-based. So Adam and Eli actually went to the same slaughterhouse, get every single pig parts they can get, from liver to pancreas to kidney, and also actually blood and blood clots and everything. And then what they found is that we found that if you have a one cycle of ultrasound pulse, and if your negative pressure is in the window of 26 to 28 megapascal, which is more than 200 times of the atmosphere, then we can reliably expand nanometer gas pockets to 100 micron within a few microseconds. That is 100,000 times of expansion, then followed by collapse. So we're talking about a bomb, but microscale, okay? And then we see the video here. So this video is taken under microscope using an ultra-fast camera, so the activity you're about to see happens within 100 microseconds, and then slow down tremendously for you to be able to visualize. So what we're seeing here, those white structures here, are breast cancer cells embedded in a clear tissue mimicking gel. And the black structures here are microbubbles generated, but they coalesce into one big one. So what you see here is that when the bubble expands, you can't really see the cells in the middle, but at the periphery, you can see when the bubble expands, cells get squeezed. When the bubble collapses, cells really get pulled apart. And if you have enough force, you really pull cells, bisect cells, as I showed you in the histology earlier. So really, it's this bubble cell interaction generating high local mechanical strain and stress that pulls cells apart. And then in a very local way, if you are out here where no bubbles and you do not see that kind of cell damage. So now we solve the two major issues, equipment and the mechanism. And I want to talk about the wonderful discovery later, which is histotrypsin stimulates the immune response, which is what I did not anticipate at the beginning. And it was a wonderful discovery along with Dr. Cliff Cho, who is a cancer immunologist. So what we found is that histotrypsin is a very unique interventional technology, unlike radiation, use ionization, or ablation like radiofrequency ablation or microwave ablation, which are the form of ablation technology using heating, right? Different energy, even using laser or ultrasound. So both ionization and heating, they kill the tissue by denaturing the protein. Therefore, you kill the tumor antigen. With histotrypsin, we disrupt the cell membrane, allow the tumor antigen to come out, but they are still functional. So then we do the first thing, which is release the tumor antigen to the body, which is critically important. Why? Because the cancer, they do two things really well. One is that the body immune system should kill any foreign things that are attacking our body. But however, the cancer, they actually help turn off the signal pathway so that the body cannot just kill them right away, and that checkpoint inhibitor therapy that won the Nobel Prize is supposed to tackle that problem. Then the second thing is the cancer hides really well. So the body can't even detect them. Therefore, a lot of the cancer do not get diagnosed until really late. Here, when we disrupt the cell membrane, they can no longer hide. The tumor antigen come out, and the body see them and said attack and tag, right? So that's the first thing to trigger the immune response. And second is histotrypsin treatment causes immunological cell death, which is something that we can spend hours on. But things like necroptosis, apoptosis, and ferroptosis, that's another thing in the immune cycle. And third is inflammation, which is actually part of the immune response too. So a cascade of those events together allow the histotrypsin to stimulate the immune response. And in this figure showing in comparison to radiation therapy or radiofrequency thermal ablation, histotrypsin induce so much more CD8 plus T cells. To the degree that we actually perform the experiments that with a mouse, implant 2 tumors and treat to only 1 tumor. but we monitor both tumors. And being able to see that historytrypsy actually stimulates CD8 plus T cells in both the treated side and also the contralateral untreated side as well. And interesting enough, this only happens if both tumors are of the same tumor line. If you have two different tumor type, right? Imagine you have one side a liver tumor, the other pancreatic tumor, then you wouldn't see the same response at the contralateral untreated side. But if they are of the same tumor line, to the degree that this is the experiment, we actually use a melanoma just because it's a tumor that is dark, so you can see the color. So this is a mouse where had pulmonary metastasis, means I have metastasis over the lung. We only treat a primary tumor with historytrypsy only, nothing else, and then you are able to see that the metastasis reduced significantly due to the immune response generated by historytrypsy. And we were also so surprised to see that this actually also seen selected patients as well. And now there's this research ongoing and hopefully lead to clinical trials soon is to combine historytrypsy with immunotherapy to enhance their effects. There are things we still need to adjust on the historytrypsy parameter side to really maximize the immune response, but this will elevate the treatment from just a non-invasive local ablation to a systemic cancer treatment. And that would further increase the impact. And another exciting thing about this is that we found that historytrypsy can stimulate immune response, right? We showed not just local, but it's systemic. And also not just in what people characterize as hot tumors, that responsive to immunotherapy. It actually stimulates immune response in cold tumors that's immune resistant to immunotherapy. So that will really expand and increase the patient population that can really benefit from actually getting cancer treatment utilizing our own immune system. So all that research is able to actually result in this FDA-approved Edison system that you will see later. And you will see the patient video and the demo. So I will end this with having a professor moment for a second. I like to think about that modern medicine, the revolution of the modern medicine is really all kind of generated, all really fundamentally pushed by innovation, technology innovation. 1860, when anesthesia was invented, that's the first time the surgery was made possible. And that made a huge impact on medicine, and that's a start moment of the modern medicine. 1900, Madame Curie discovered radiation. And that enabled radiation therapy in 1910, where you start seeing that patient can actually get treatment from radiation, even though it's toxic, but it can actually treat tumor without, doing the surgery. 1960, really big revolution, technology innovation from optics, where the endoscopic and the laparoscopic surgery become possible because you can go in with a small lens, allow you to actually see target inside. So you don't have to perform surgery by opening really the whole thing. You can do small incisions and go in to do laparoscopic surgery or endoscopic surgery. So every 60 years, there's a big technology innovation that now really change how medicine is practiced. It's another 60 years. The time is right now that the medicine whole movement is moving towards minimally invasive, non-invasive. And it's my belief that the next era of medicine is entirely non-invasive surgery. And it's starting from historytrypsy. And not just liver tumors, renal tumors, and pancreatic tumors. We're looking at different targets also from breast tumors, prostate tumors, thyroid tumors, anything you think of. Now we can actually treat through the skull, even into the brain, not just the tumors and also neurological disease and the stroke. So there are so many things historytrypsy can do, but today you will see a clinical Edison system here to start with actual clinical patients with liver tumors and see how it's in action. Thank you.
Thank you so much, Dr. Zhu. And we started this afternoon with a panel who were discussing deep tech and the 25-year journey from discovery to deployment in a market, and it was remarkable to hear about your 25-year journey just now. Congratulations on your perseverance and the incredible outcomes you've achieved. To continue from Histostronics, we've got Josh King, who is the Vice President of Marketing and Global Market Access at Historsonics, where he leads global marketing strategy, brand development and market expansion initiatives to accelerate the adoption of the company's breakthrough Edison histotripsy system. With extensive experience in healthcare marketing and commercialisation, Josh has built his career at the intersection of cutting-edge medical technology and patient impact. He specialises in translating complex clinical innovations into compelling value propositions that resonate with physicians, health systems, payers and patients. Prior to joining Historsonics, Josh held senior marketing roles in the medical device sector, where he successfully led product launches and international market entry strategies. His expertise spans strategic marketing and global market access, with a strong background in biology and medical physics. Please join me in welcoming Josh.
Sure. Good afternoon. This is on. If you don't know Professor Zhu, you do now. She's a force of nature. And if you are part of an organization looking for founders, she is the model you want. She's unstoppable in almost every way. I see the people who know her well laughing, because I've been lucky enough to be around Jin for almost 8 years with Historsonics, leading the marketing team. I think we're going to do this, and normally we wouldn't do what I'm about to do, but we've talked about the science, we've talked about some of the immunology and work. Even though we're not approved in Australia through the TGA yet, we're finding every way to get in. We do want to actually share with you something that I hope hits very close to home with you, because it does to us at the company. We're going to share a quick few-minute story from a patient actually from Australia, Kate, who flew to the US to get treatment because she had no other options. And what I want you to hear in her voice is the desire to have hope, why she needed something. You all are working in here to advance science, to advance medicine in a certain way, and I hope that you have the same experience that I and my colleagues have had. So I'm going to be quiet and have Kate talk to you for a few minutes, and then I'm going to tell you a little bit about the system technology, and then the most important thing we're going to do today is we're actually going to show you a demonstration of this live. So bear with me, and folks, hope you can start Kate.
So my name is Kate. I live in Perth, Western Australia. I'm a mum of 2 and I'm married to my husband Charlie, and I love spending quality time with them. I really enjoy baking because I love to teach them things, and I just love food. Back in 2019, I was 35 years old and I was breastfeeding my daughter Grace when I felt a lump. I had a bad feeling, so I went to the GP to check to see what it was. After a bunch of tests, I was diagnosed with breast cancer, estrogen positive, but at that point it was stage 1. I opted for a double mastectomy rather than a single. The surgery itself was very intense and was a long recovery. But once I got through all of that and the chemo started, that was when the sickness really kicked in. I had a good 2 years where I was deemed in remission, and I went in for my appointment to see my surgeon just for a checkup. I thought that I was going to be okay, but unfortunately it had metastasized into my sternum, which considered me a stage 4 cancer patient. Back on chemotherapy, which is something that I really did not want to do, considering the past and how hard it was to go through all of those side effects. But I went on a clinical trial and tried some estrogen tablets, like resistant tablets. That didn't really do what it needed to do. So it continued to spread into my brain, my spine, my lymph nodes, and my liver. So my options were starting to get limited. I first heard about histotripsy from a friend who introduced it to me, and I started doing my own research to see if it was an option for me. I didn't really have any concerns with histotripsy, knowing that it's non-invasive and that it's day surgery, there's no incision, and it doesn't actually damage any of your healthy tissue. So I thought it was a great option. Straight after the procedure, I felt good. It was very nice to hear that the doctor had actually targeted 5 tumours successfully. When I got back to the hotel, I did get up and go for a number of little walks, and I was really surprised at how good I felt after the procedure. I can't believe that there is technology out there that can target tumours directly without affecting your body, without any side effects, and you can literally get up the next day and go about your normal life. 6 months after my first treatment, I decided to go back to the US for a second treatment, knowing this time what to expect. I woke up and I felt better than I did the first time, because he targeted 3 tumours on the left side of my liver, and I had absolutely no pain whatsoever. When my radiologist saw the scans, he was pretty impressed with how my liver responded to the treatment. I would love histotripsy to be available in Australia, and I would like more radiologists and oncologists to know more information about histotripsy, as I think it is a very powerful treatment option. I understand that data is important for my medical team, but as a cancer patient, I don't have time to wait years for data to be collected in order for them to feel that it's my time to try this technology. What inspires me to keep pushing? Definitely my children. I do really want to see my daughter and my son maybe get married one day, and I have a strong feeling that I'm gonna get through it. I don't want to go anywhere. I love them so much.
You've got Jinju. You've got patients like this who have no other options, and that drives our company, it drives our philosophy, it drives everything we've done. I don't have the timeline slide of 25 years on the timeline, but we do like to say it's a 25-year overnight success. So everybody that's a founder or CEO of a company that you started, I'm sure nothing happens as fast as you like. I'm going to talk a little bit about the company and the backdrop of the patient, the backdrop of the partners we have as investors, clinical partners, advisors, really shape the way we consider what we do at the company. So our big vision is to basically accelerate the opportunity for procedures that are done today to be done non-invasively, not just for tumors, definitely not just for tumors of the liver, which we'll talk about why we started there. As you've heard, tumors of the kidney, the pancreas, the brain, the breast, thyroid, prostate, sarcomas, metastasis from all of those as well are totally in our sights right now. Some of the things that you may not hear talk about, things like hypertension, metabolic and neurologic diseases. Because we can transmit this phenomenal new energy source through bone, through the brain, without making a decision, we see this as an opportunity that's really going to advance science and medicine across multiple disease states and application. We're going to start here, where I'd like to draw your your eyes to that bottom ribbon from palliation to cure. I don't know about you, but when I think of palliation, I think of it 20 years ago when I first heard the term when I was volunteering in hospitals. And it was kind of like, it was toward the end, you were just trying to make somebody, feel less pain as they left us. That's changed now. Palliation is a lot different. We've seen with patients who've received histotripsy who have had extreme pain from pressure pushing up against their liver capsule or on nerves. They get histotripsy, pain goes away, and they go home that same day. They go for multi-mile walks with their family they hadn't been able to do in years. Palliation's a different thing now. If you think about the number of patients that are surgically operable versus not, the ratio is generally about 15%, 20% to 80. 15 or 20% are surgically operable. The patients that are getting histotripsy right now don't necessarily fall in that because we don't have data to compare it to surgery. We hope to one day. But the population of people who need their tumors removed, I think if you added up all of hers, we were 10 or more over a couple sessions, without toxicity for her, without an incision, without the complications of surgery, allowing her to fly back to the U.S., fly to the U.S. and back twice and be with her family. Stand alone, if that's all we did as a company in the liver, we'd be happy. The market is phenomenally huge, but we're going to take it on to a new level. We think this is going to make more patients eligible for local regional therapies with both curative intent as well as palliation. What you're going to see in this demonstration we're going to do, if you're a surgeon trained here versus a surgeon trained in New York versus one trained in Hong Kong, the way you do the exact procedure is going to be different where you're trained. Histotripsy removes a lot of the art. We respect the art for sure, but this allows us to standardize and democratize this procedure. So if you know how to look at the liver under ultrasound and CT and MRI, we can train you to use this. Multiple specialties can use this, and the outcomes, the energy, should all be very, very similar. So we think this is going to introduce a new opportunity to collect data in advanced science as well. The economics of this. We have, through great effort from our colleagues here, we have a system that just started doing procedures in the UK. The UK has a queue of patients lined up to get treated for major disease conditions, but they can't because the beds are full. They don't have enough staff. It's the same story everywhere. Well, they started using histotripsy as part of an accelerated pathway, and they're hoping to reduce the queue because they can get a patient treated and potentially send that home same day. About 98 or 99% of all patients with histotripsy go home the same day. Not on narcotics. If they need any pain relief at all, it's over-the-counter stuff like NSAIDs. So really redefining the economics of patients moving in and out of care and being able to get 1 or 2 or 3 or more tumors treated in the same session totally revolutionizes the way you think about patients coming into a hospital and going back home in a productive life. So our solution, I won't beget this anymore, but to liquefy and remove tissue. Removing tissue is one of the key words. It's not desiccating the tumor like you would if you had heat, risking burning something. It's not ionizing radiation, which knocks off electrons and destroys the tumor, hopefully, in place. This actually liquefies it. The one thing I don't know if we talked about is what happens to this liquefaction? What happens to this lysate? It's processed out of the body over weeks and months. You can see it radiographically. It goes away and shrinks, and I'll show you what that looks like in a second. This is our solution, and we think we're going to change the paradigm in non-invasive, very low, if not non-toxic, low complication procedures for patients across conditions. And you can imagine that's why, as you know, we worked diligently with our investors since I joined about 8 years ago. This is the shared vision, and we've only accelerated that with our new partners. The problem we're attacking right now, as mentioned, 4 out of 10 of us in this room are going to develop some sort of tumor that's cancer-related. Not even talking about benign tumors, which would make it even more. 40%. I can ask the question if you know somebody in your immediate family or a friend that is battling some sort of tumor right now. My guess is 100% would raise their hand. They don't have to, but we've done this enough, we know. And it's horrible. It's a horrible condition, and your options are more limited than maybe you think once you get into medicine. More bad news. In the US, it's going to grow by 50% in the near future. It's just a fact, our way of life. It's what we do. We look at these numbers all the time. We're constantly evaluating, hoping that that number comes down. Unfortunately, it's not. Globally, the the story's worse. As countries become more sedentary, the diets become more westernized, restricted, where people don't eat right, the numbers are even going to climb higher. And that's going to be a challenge for all healthcare systems. And we know that, and we think we have a solution that can maybe penetrate that as well. So we are approved in the United States, and a handful of other countries I'll show you in a little bit, for the destruction of liver tumors through the mechanical mechanism of histotripsy. And we started in the liver. People ask us all the time, why in the liver? Why didn't you start in breast or something like that? Every organ or disease condition you see on this holographic person here all shares a blood supply to the liver, some form or fashion. Some really direct, like colon and rectum tumors, direct pipeline to the liver. As tumors escape their original primary origin, they go and they seat themselves in the liver, and they become a real problem. Many patients don't die of their primary tumors. They die of overwhelming liver tumors that make normal liver function impossible, and they end up passing away. So not only do nutrients and toxins get filtered through our liver, but every tumor that goes throughout the body and the blood can find a home in the liver. That's why multitudes more metastasis arrive in the liver than primary tumors arrive. If you have flown over here from Asia, right? Between just a handful of Asian countries, half the world's liver tumors occur in just a handful of countries over there. Right? And right now it's a tremendous problem, and we aim to solve that as well. In relation to this, if you have cancerous tumors, over some point in time they will metastasize to your liver, and that will be a direct reason for your mortality. That's how important the liver is. It's one of the only organs you can't live without. Even if you do get treatment, you get it resected, you get it ablated, you get a probe stuck in, heats that tumor up to 100 plus degrees Celsius, cooks it, or you get radiation, 60 to 80% or more of those can come back. But you only have so much liver. You have only so much health that can allow you to go through some of these really highly invasive morbid procedures. Histotripsy, as Professor Xu explained, can be done over and over again, preserving healthy tissue. Right? This all sounds too good to be true. This is the point in the presentation where I say, when we get to the demonstration portion, I'll ask you to suspend disbelief, because I think what you're going to see is going to not seem real, but we're going to show it to you in real time here. So our solution is histotripsy. In true Michigan fashion, this little video that's playing is a block M. If you look at the axes on this, it says 1 centimeter on the top. It's also 1 centimeter on the y-axis as well. This is steered electronically in the form of an M in a medium to show how precise this treatment can be. 1 millimeter outside of where this is designed to go, it's perfectly safe. There's no radiation hitting it, it's not burning it, but inside it's complete liquefaction, as Jin said. Instantaneous cell death in a volume that the physician can plan and watch in real time, just as you're seeing here. We do not carve letters in any organs, just saying, that's unethical, we don't do that. Here's the system. This is the Edison system. We named it Edison because Thomas Alva Edison actually had strong Michigan roots. He's the most prolific inventor in the United States, maybe worldwide, holds the most patents. Roots in Michigan, and we thought, you know what? We want to make the most innovative technology that meaningfully impacts the patient's lives around the world, and this is it. It's got everything in this cart is mobile, which means you can move it from room to room. It doesn't have any special shielding. You can plug it into a normal wall outlet, as opposed to what Jen did with her system, where she had to get some sort of nuclear generator to power the original one. We have a complementary ultrasound system on the left that powers direct visualization in the tumor as well. So integrated imaging with therapy in a mobile device that can be shared room to room across multiple specialties, treating multiple organs. I won't belabor this. The picture you see here is a patient that was in the Hope for Liver trial, had a 2+ centimeter tumor right in the middle of that black spot. This is within 24 hours after the procedure. This is a scan of that patient. You can no longer see the tumor, and all that is liquefied. Within 2 months, that completely resolved, and that patient had you couldn't tell that patient had any treatment done to them at all. The only thing that says that they had it done was this image post-procedure, and that patient walked away, and that tumor did not come back. Here's another one. When I said, what happens to that lysate? What happens to this liquid that you create? On the left is a 36-hour post-treatment image. You can see the difference in the white area and the dark center of what looks like almost the perfect sphere. And then with 1 year later, it's just a very little, almost indiscernible little mark, maybe a small bit of scar tissue there that you can see where it was. No tumor to be found. Again, if you look at the skin, the subcutaneous tissue, no damage to any of that as well. Evidence. This was mentioned a little bit. I won't go through it, but if you look at what we are currently funding as a company, and you can see the icons here, liver, kidney, pancreas, and prostate, we are basically in this active enrollment area about to move into the future where we're going to be studying the effects of histotripsy for safety, short-term efficacy, and long-term efficacy. So if you're a scientist or a physician and you like your 3 and 5 year data, we're working on it. We can't accelerate time, but we're capturing every patient. This Boombox trial is a prospective post-market study where if we develop a partnership with the hospital, we capture the details on every patient you treat that you can have. We share it, looking for signals where we can develop powered hypothesis prospective trials to look for signals, both in efficacy, immunologic outcomes, everything. So we're really funding our research to make this a really powerful tool going forward. The 1 year data that we're past this now, but this was a pretty pivotal 1 year follow-up in our trial, our Hope for Liver trial that was done across the U.S. and Europe. What I'll show here as you kind of look at the numbers, a 90% tumor local control rate. 90% control rate at 1 year. If you don't have a perspective what that means, it is comparable to every local regional therapy that's been out for 20, 30 years. Except in this patient population, there were primary liver tumors, metastatic tumors, and every patient had to either fail 1 or 2 lines of standard of care therapy or be intolerant of that therapy for some reason, health-wise or whatever, and they underwent histotripsy. So we had a very sick, aged population, and we were obligated to perform safety and efficacy in that population. You can see over the right side, the survival, 73.3% at 1 year with HCC, and METs, obviously a little lower, but still very comparable. Interesting enough, there's not one physician in the study that performed a procedure that had ever done histotripsy before. So it was the first patient was enrolled, and we followed them all the way through. So the novelty of this technology, we built it into a system that we think we can train and get people to use it very quickly and be experts very fast, right? And we continue to advance the technology. So if you are adopted, we train you, stay with you, accelerate you through that learning curve so you can treat as many patients as possible. I'm getting close to the end of the presentation. If you have your phones handy and you like to take pictures or videos, I'd say get ready to take it out before we start the demo because there's going to be a video that you probably want to go home and show some people. But we are probably close to 3,000 patients. But what I think is interesting is this activity by specialty in the middle. Almost half is surgeons, almost half are radiologists using it. There's a radiation oncologist as well using it, and then even a hepatologist. But the split, right, the number of physicians that are trainable to do this really opens up access to patients. And we feel that's phenomenal. And we have a world-class training staff as well. The liver segments, we treat every part of the liver. If you've ever been around liver treatment, tumors that are really high near the diaphragm or heart are very difficult to treat. As Jin showed earlier, tumor that sits right in between a bifurcation of vessels, you don't want to go sticking a needle. You don't want to go in there with surgery. And if you hit it with ionizing radiation, you have a chance to hit the heart or lung. Histotripsy has been phenomenal in those procedures as well. So every segment of the liver, and you can see the bottom row here, tumors treated by the tumor type, colorectal mets, Almost everybody that has a tumor in their colon or rectum, it will metastasize to your liver. And so we treated a ton of those, and you can see HCC is hepatocellular carcinoma, that's primary liver tumors, and you can go on and on and on. There's no mutational specificity that histotripsy can't treat. We can treat every tumor that shows up in the liver, right? And that's something that's also very unique. Here's the US map, just to show you a lot of people. What's not embedded in this is 8 out of the top 10 cancer centers in the United States all have histotripsy machines, and most of all those are in active research as well as treating patients clinically. Globally, our new partners with Horizons and others look at this map and say, why can't we go faster? So we're going as fast as we can. I think we're really excited about making a difference in areas where the need is dramatic. The one thing you don't see here is Australia. As I said, we're working diligently to try to get through the TGA. We're working, aligning ourselves with your experts here to really make a difference. So we're excited about this number. If we come back a year from now, this number is going to be drastically improved and changed. Going forward, the top row, this platform 1, you can see liver, kidney, and pancreas. Think of that as where we're at today. We're progressing fast. Prostate platform for BPH to start is going to be a world changer. Think of a totally non-invasive way to destroy and liquefy benign prostate hyperplasia, get rid of lower urinary tract syndrome, hopefully, and move patients into a totally non-invasive way to overcome a really deliberate, problematic situation for you. Sorry. Neuro, as I said, not just tumors, but think of intracerebral hemorrhage, destroying non-invasively liquefying clots in the brain so they don't have a cascade of effect and cause problems down the road. Application extensions, thyroid and breast, right? Technically, these are really probably pretty easy for us to do. We're just trying to assemble the right teams to put them on it and let them run fast so we can non-invasively attack those disease states. Finally, abdominal, uterine fibroids is a huge worldwide problem. What if you could non-invasively, mechanically destroy those in minutes and have patients go home the same day, right? The goal of that is within our grasp, I think. And finally, neuro and spine, right? Many patients who get tumors outside the spine, particularly brain, will metastasize, or lung will metastasize to the spine, and there's not a great solution for that other than surgery and radiation right now. So I've asked Bella to come up here. Bella's one of our engineers. She's phenomenal. She's going to start a process of getting the system ready for this demonstration. So if you've got your phones handy, get ready. She's going to put it in there. She's going to get it about 40% of the way to plan. Before we switch over to a different scheme on the TV, I'm going to go through a quick video, and I'm going to show you what this looks like on a patient. So you have some context to what she's going to do in this water tank here. This is what comes with the system. As you see here, a third-party imaging source is piped in so we can see live ultrasound. The generator itself, the robotic arm, and a therapy transducer that sits at the end of the robotic arm. We know exactly where the cavitation is going to occur, right? So we know, and we line it up with a diagnostic ultrasound transducer so we can look at it in any plane. And then finally, we have a little cart here that makes degassed water. That allows the propagation of the ultrasound force totally through the patient into a tissue-transparent membrane that sits on the patient, dampens the movement of the patient, and allows us to send the signal through unadulterated. Again, actually a modern marvel here. So I'm going to hit play on this video, and it's going to walk through some things. So the physician controls the robot through this space mouse, 6 degrees of freedom. They will lower the therapy head onto the patient, which sits inside this single-use disposable membrane that's completely acoustically transparent, and that allows us to both see and treat the tumor without losing fidelity of the ultrasound signal. So we target it here. The physician controls the tumor treatment plan using ultrasound as well as MRI or CT combined, fused real time. Once they develop the plan, the physician just hits the automated treatment button. That's it for touching the system. The treatment arm will then begin at the deepest point and raster through the treatment volume, as you see it here, with submillimetric accuracy, starting at the deepest part of the tumor. and it's going to go through the planned, not only in the tumor, but the physician plans a little bit extra space outside of that called safety margin. That's to make sure no little small cells are outside there hiding. It goes from the bottom all the way to the top, overlapping itself 50% every time to make sure no tumor cells escape. Once it's over, the physician just simply lifts it off the patient. You pull the coupling kit aside, and the patient walks out almost exactly like they walked in. After probably a couple hours of observation, most patients go home. Making the impossible possible is actually a quote from Jinju as well, and we live by that. So now that we're almost ready, what you see here in the right, this is a piece of beef liver that's been cubed. This is a piece of everyday beef liver cubed up, and it represents a tumor for this demonstration, okay? It's a solid piece of tissue. Outside of that, it's in a gel phantom that's very, very sensitive, right? So what we're going to do is we're going to put this in, and what Bella's doing right now is she's taking pressure readings, basically, to determine how much energy throughout this whole phantom, throughout this tissue that is required to destroy it. So the physician will dial up 7 different points to decide how much energy is required. Once you hear the sound, by the way, that sound you hear is actually the sound of bubbles forming and collapsing inside the patient or inside this phantom. So we can hear the treatment being active, and we can also see it, and which you're going to see on the lower left. So she's created her plan. She's taken all 7 points. What you get from that is a planned treatment where you have all the information and you have the time. What does that say, Bella? 4 minutes and 9 seconds. So the physician, when they plan, they say, no, I'm going to be sitting here for 4 minutes and 9 seconds watching myself destroy this tumor in real time. So if we're all ready, Bella's going to hit play, and the treatment will begin. You'll see it dive down to the bottom, and you'll see the bubble cloud. If you look under that red crosshair in your bottom left image, the red crosshair, that's where the bubble cloud is. You can see that white hyperechoic area, and it's going to be rastering through. You can actually start seeing it liquefying this tissue in real time right here. What you see here actually mimics what you see here in real time. So the physician can watch in the green, that's where the tumor's been treated. The orange is actually where the bubble cloud is in active space right now. So we joke, this is the time when the doctor goes and gets coffee, and they allow someone else to see the procedure. I'm joking. They stay there for the whole time. But they can observe what's going on. I'm going to go back to a little bit of the physics of what Professor Xu said. The bubble clouds are expanding and collapsing from nanoparticle size to multiple times the size of the cell, collapsing around it, tearing that cell membrane apart, ripping the subcellular anatomy apart. So even if you look at it on cytopathology, you can't tell exactly what it is. You don't know. It's completely liquefied and acellular. There's no heat here, right? And if you know about heat in terms of organs, it can kill, and it can kill tissue fast, and that's great. However, it's uncontrollable. We call it thermal chaos, right? When you heat something up, if there's a blood vessel that runs by it, there's a good chance you burn the vessel. There's also a good chance the blood pulls heat away so fast that it doesn't have time to kill the tumor, and you're left with viable tumor there. So thermal's been around for a long time. It's fantastic for a lot of procedures, and they've proven a lot of safety and efficacy with it. But histotripsy does more. It does more without that thermal chaos. So we're 2 minutes and 15 seconds left. They're calling the doctor back in the room to see how it's progressing. If you can see here, what was this cube piece of tissue now looks almost like a perfect egg. When I talk about precision, what's killed out here at the periphery is the same that's killed in the middle, right? So uniformity of tissue damage. All this information that you see on the screen right now happening in real life to a patient is recorded, right? So we can, in the future, we'll know exactly what parameters went into treating these patients, how they did acutely after the procedure. Plus, in the future, we can look back and say, wow, this person had this type of tumor. They got treated for this long with this volume. And here were the complications they had. Here was the outcome they had. Potentially in the future, how did they perform when on an immunotherapy that was combined or chemotherapy that was combined? So now you have a totally non-invasive sequence of treating patients over time that you can follow, and you can start dialing in exactly when and when not to provide systemic agents if they need them at all. Again, we're preserving tissue. What you don't see in here is any damage really from here to here. That's because the energy is coming in. So Bella finished the procedure. She's going to pull this out. She's going to put it on this table, and we're going to refocus the camera. What was formerly a solid piece of cubed liver tissue, we're going to show you in real time. It's no magic. It's magic of science. We're going to show you exactly what that looks like. She's wiggling it out of the holder there. No pressure, Bella. So if you're not in the front row, you can look up here. We're going to zoom in on it. And she's going to do what every surgeon does, is she's going to cut right down the middle. That lysate is now acellular. It's going to get resorbed and expelled out of the body naturally over the course of weeks and months. Every time I see it, I get the chills. I can't believe this happened. This technology is new to the world, but we're sharing it everywhere we can. This is histotripsy. I ask you to suspend disbelief when you see what you just saw and think about the applications in the world that we can make a difference in. I want to thank University of New South Wales for having nothing but the hospitable environment for us. It's been very welcoming. Thank you so much. To the Horizons Venture Group, Selena and her entire team, thank you for believing in us. You're amazing, every bit of it. If you're starting a company, I hope you find somebody like the investors that we have, and I hope that you all have success in your field. I'm happy to answer questions after this if you like. Thank you very much.
Thank you so much to the Histosonics team. That was quite incredible. We now have a moment for a very special gift announcement, and we have 3 speakers who I'm going to invite to the stage individually. Our first speaker is Senator Dave Sharma, who has served as a Liberal Senator for New South Wales since 2023, and he's currently the Shadow Assistant Minister for Competition, Charities and Treasury. He was previously the member for Wentworth from 2019 to 2022 and has contributed to parliamentary committees on social media, online safety, industry and innovation, the National Redress Scheme, Community Affairs and Environment and Communications. Before entering politics, Dave has a distinguished diplomatic career, including roles as Australia's Ambassador to Israel, Assistant Secretary at DFAT, and the head of the International Division of the Department of Prime Minister and Cabinet. He holds degrees in Arts and Law from the University of Cambridge and a Master's in International Relations from Deakin University. Dave also brings experience in technology and enterprise, having chaired Israeli tech firm Shekel Brainway and served as a director and partner in private sector ventures. Let's welcome Dave to the stage.
Good afternoon, everyone. It's very difficult to follow in terms of content or engagement what we've just heard from Professor Zhu and Joshua as well, but I wanted to congratulate the team at Histotripsy and of course the team at Horizon Ventures who've chosen to back them, because I was expecting to come here and talk to a few people, hear a few interesting things. I didn't expect to be blown away in the way that I was by the promise of this new technology, and the story you had from the woman in Perth who travelled to California was quite moving. So you've given me a project for next week, which is to go down to Canberra and find out what the TGA is doing and why they're taking so long. And when we can get this approved. I don't want to speak long today. I did want to acknowledge the Chancellor, David Gonski, who's here, who's a great supporter of the university and indeed a great supporter of research in Australia, of business, of philanthropy, of academia in so many ways. And it's a real an asset that the University of New South Wales has Chancellor Gonski as their Chancellor. I just wanted to talk a little about the theme of today, though, which is the importance of innovation, research and commercialisation to address some of humanity's most pressing challenges, whether it's the fight against cancer, which we've just been hearing about today, whether it's the fight against other non-communicable diseases or communicable diseases or the diseases of degeneration and ageing, whether it's the significant environmental and climate challenges we face as a planet, whether it's challenges that have been with us for millennia, really, the challenge of poverty, of hunger, of disadvantage. It's innovation and research and the commercialisation, application of new ideas and technology that is really the story, the underpinning, the engine of humanity's progress throughout our history as a civilization. And it's the spirit and the methods of inquiry and debate and research and discovery that institutions like the University of New South Wales support, that has driven that journey. And when these spirits, these methods, these institutions have been suppressed, the spirits of inquiry and discovery and research and debate, humanity has suffered. And when they have been supported and nurtured, humanity has moved in a century as far as we've moved in the millennia preceding it. So institutions like the University of New South Wales really embody and catalyse and champion and nourish that spirit. And in many respects, it's the health of our universities that correlates with the health of our society in the broadest sense. So I wanted to just pay tribute to the University of New South Wales today, but also to Horizon Ventures and Selena Chow and your co-founders, because you are a key part in taking these great ideas, the great ideas of people like Professor Zhu, who've been toiling away in laboratories and research centres for decades and years, underpinning it with the capital that's needed to advance these technologies, to scale these technologies, to conduct the clinical trials, to take them to market, and to ultimately ensure that humanity benefits from all of them. I know we now have a special announcement, on which I don't want to proceed or preclude, but I just wanted to start by recognising the work of Horizon Ventures, the work of UNSW, and again to commend Professor Zhu and the entire team at Histotripsy for the tremendous work you've just showcased to us today. Thank you.
Now it gives me great pleasure to introduce our Chancellor, David Gonski AC. One of Australia's most prominent business leaders and philanthropists, David Gonski has been a formidable advocate for education, the university and its mission. Mr. Gonski has a long association with UNSW. He is the first UNSW alumnus to hold the position of Chancellor, elected in 2005. He is also the chairman of the UNSW Foundation, and his role has been absolutely critical in building the university's philanthropic culture and support. Thank you, Chancellor.
Thanks, Bronwyn. Ladies and gentlemen, firstly I want to acknowledge Selena Chow and also her partner, Debbie Chang. Without you, we wouldn't be here today, and without you, we wouldn't have seen an incredible presentation together with all the other things we've seen. So thank you, and I have to say, he deserves it. A Chancellor's life is quite a dull one until I met Selena 2 years ago, and she and Debbie, we chatted, and I've got to say I've still been working on all that fuel and enthusiasm you gave us, and we thank you very much for it. Ladies and gentlemen, if I may, talking about enthusiastic and wonderful people, I also acknowledge Dave Sharma. It's wonderful for him to be here, And I've got to say, having him as one of our senators in the big smoke of Canberra makes me sleep much better at night. So thank you very much, Dave. As I sat through today, I had this mixture of emotions. First, an enormous pride that this happened at the University of New South Wales. Thank you to all of you for coming here today. Thanks to those who put it together, and thanks to Selina and indeed Debbie for allowing us to do this at the University of New South Wales. This was then mixed with hope. I mean, if I'm going to have one of those tumours, I really want to have histotripsy. So if you don't get it through, Dave, we'll have to all do a deal through the airport, which I have an interest in, and we'll be off to America. So thank you. Then there was envy, that wonderful buzz of the entrepreneur, of the founder. Congratulations to each and every one of you who performed today and showed us what really we should be envious of. People in Australia wanting to do things, absolutely marvellous. And I should mention people doing things around the world as well, also fantastic. But it was mixed, I have to say, with a bit of sadness. I'm not a founder. I don't have that get up and go to do. And finally, I've got to say, I still think that LLM is a master of laws. So you can imagine how I feel, and I might say I'm the only person wearing a tie here today. Ladies and gentlemen, because time is moving, let me move to the secret announcement that was on page 13 of the Sydney Morning Herald this morning. It is an enormous pleasure on behalf of the whole university to announce a transformative donation of $30 million from the H.S. Chow Foundation. This generous donation came from an idea that Selina and Debbie had as we walked round 2 years ago. They said they would do it. We, of course, wondered. 2 years later, they've delivered. And what are they delivering on? Well, we're going to build the New Horizons building using this money. And as part of that building, we're going to establish the H.S. Chow College. Now, this college will be unique for this university because it'll be a home for female STEM students. It'll nurture leadership, collaboration, and a strong sense of community. And in my opinion, and I've only been here 20 years and 4 months, we really need this. And this is going to bring us into line with other international universities. Thank you sincerely to the foundation, to Selina, to Debbie, and to all that have been involved. We appreciate it. And the wonderful thing, just as you sit there watching the wonderful things that that machine did, come back in 10 years' time and see the wonderful graduates who've been through that college of yours and who will think the college changed their lives, just as somebody said that Baxter College changed theirs earlier today. Thank you for being here, and I think it is a great honor for me to call upon Selina to talk to us. Selina.
Well, thank you. Thank you. A very good afternoon to everyone. And thank you, Senator Sharma and Chancellor Gonski, for your kind words of introduction. It is a privilege to be considered a friend of the university and a friend of New South Wales. So, myself, in 4 words. I'm a Christian. I think I'm a learner. I'm a traveler and an enthusiast, as you can see. And a one-liner about our company, Horizons Ventures. We are hard at work advancing science and technology for yield and impact. So, one word for my time with New South Wales University, pivotal. Pivotal is the word that always springs to my mind when I think back to my days in University of New South Wales. Coming to Australia was my first adventure beyond the city of Hong Kong, and Sydney's sheer vastness made the world felt both wonderfully inspiring and humbling. Between the wide open spaces and mental space of the university, I somehow found myself reading and rereading Don Quixote, a book that recalibrated my perspective on what mission is, what competence is, and what service is all about. It changed my life. Perhaps more than any lecture. Okay? Was Cervantes' famous line, "I know who I am and who I can be." Well, in the modern day business MBA courses it is a classic Dunning-Kruger effect and remains a constant reminder for every aspirant how pitfalls of misperceptions can complicate the present and cloud your future. It was here at the University of New South Wales, surrounded by Australia's special brand of practical idealism, that I began shaping my own philosophy and outlook in life. So it was very simple. Before deciding who I want to be, I must first understand who I don't want to be. Only then can I begin to imagine who I ought to become. This insight continues to guide my quests and perspectives, whether the improbable can be achieved through venture investment, and if so, how? Can I become the most able enabler for myself and for others? I remain profoundly grateful for the opportunity to be part of UN's University of New South Wales and your ongoing story. So please accept this heartfelt gift from me and Team Horizons as a token of our deep appreciation for the university that helped me find both my direction and perhaps myself. I want to end with sharing with you all that last week I bought a new sculpture for our office, a Swarovski-cladded, gold-plated dinosaur skeleton. As a reminder to myself, if I lose my open mind and don't recalibrate myself time and again, I am but a Swarovski-cladded, gold-plated dead fossil, which I don't want to be. Thank you again for joining our Techcracker event today, and thank you very much for the opportunity, your kind words, and your company. Thank you so much.
Something to take on the time with.
Yeah, thank you so much. Thank you so much. So much.
If everyone would be ready, we're just going to take a photo from the other side.
I'm posting for something, sorry. No, what I really want to be is to be a stand-up comic. Working for BBC. Done? Done? Ready? Okay. Assembly. What? Oh, okay. Take a picture.
So you got it. I'm going to be there.
We want to take a picture of all of you.
Can I encourage all of the speakers and panelists to come up and have a photo taken?